I guess restrooms are down the hall on the left, I believe. And we have supposedly a refreshment bar out there. So if you're just sitting there watching this stuff and you decide this is super boring, I would rather eat a candy bar at some point, they will be there. This is basically sort of a workshop that is built in a sort of a two-stage process where the first day involves a fair amount of didactic exposure, and the second day is mostly try it out with your own hands. So in the first day, we generally try to keep the didactic stuff going so that we don't actually run it into the second day. With that in mind, for questions, we'll have question and answer sessions at the end of each block, so before the mid-morning break. Those are the environments that are best for asking big conceptual questions. If there's a question about clarity or something that really does make sense, do not wait until the end. Definitely raise your hand, and if I don't notice you, then actually yell at me or something like that, because every once in a while I lapse into some kind of terminology that works for one part of the audience or another. What I'm going to do is tell you guys a little bit about what we're going to be doing, conceptually today. It's kind of our, if you will, our curriculum. What we'll do at the very beginning of the workshop is essentially talk about RNA and why we would bother with it in the first place, what good it does us. And when I use the word us, this workshop is targeted predominantly towards social and behavioral scientists. So this doesn't mean that the stuff that we're talking about isn't similarly applicable for clinical research or clinical scientists or people with, like, I don't know, sort of like basic RNA biology interests or things like that. But the kind of the context, the assumptions I make about what people know or what they're going to want to do are mostly filtered through the lens of behavioral science. So we'll talk a little bit about why RNA is useful in that particular context, the kinds of things people do with it. And then with that end goal in mind, talk about the things sort of, if you will, technically or procedurally that we need to pay attention to actually make RNA data useful for those particular ends. And you would have, actually, it turns out, different decisions about what to do if you had different sort of goals and objectives. So we'll be looking at this mostly, again, through the lens of kind of behavioral science, social science, that kind of thing. So we'll be talking about very pragmatic things about, like where would you even collect RNA from? For those of you who are actually doing your own studies, other people will be inheriting big data sets like this. This workshop is predominantly motivated by the arrival of RNA data in the health and retirement study. So this is, sort of a huge opportunity for social and behavioral scientists nationwide, worldwide, to actually access this kind of information without going through the suffering of figuring out how to sample, how to get the assays done, all this other kind of thing. But I know that many of you in the room are doing your own studies, either planning them or have, midway through them or have data in hand now. So I'll be trying to cover all of those interests in addition to the folks who will be sort of inheriting the end product through data distribution from places like HRS or MIDAS or AdHealth. We'll talk a little bit about how RNA gets assayed, what we're actually measuring there, in part because that ends up driving some of the terminology that you'll see as we try and deal with actual data analysis. So there'll be words like reads and read counts and normalization and stuff like that are actually helpful to know, in part because there's no one right answer for all of this. And so how you address these topics will depend a lot on your kind of personal research goals. So knowing something about what went on before you actually got the numbers you have is helpful. You obviously won't have to, make decisions and calls about this, but it's not a bad idea to figure out how, when you actually process your own numbers, those decisions might impact what you're doing and ways to actually switch out the, if you will, the world you're operating in. Right before lunch, we'll talk about what you could think of as sort of the traditional approach to analyzing RNA data, which is actually sort of a almost mindless variant of how people were analyzing DNA data. Then after lunch, we'll spend the bulk of the time talking about why analyzing RNA data as if it were DNA data is actually probably not a great idea, and to bring along some different concepts about what makes RNA data different from, let's say, DNA data or other kinds of biomarker data. Both some of the challenges that brings up, but also some of the opportunities that those sort of essentially these giant correlations among different gene transcripts can present. And so the goal in the afternoon session is to help you guys get aware of essentially sort of the mass action structure of what's going on in RNA data, so that instead of trying to fight it the way traditional geneticists do, you can actually surf its energy and, sort of essentially build insight and sort of move with nature essentially instead of against it, which is what would happen if you were pursuing the kind of straightforward approach that we described before lunch. You can think of it as like the default genetics approach to RNA. So after that, next day, we will be having, like I said, a in-person kind of practice on your own workshop. At the end of today, I'm going to show you guys a bunch of interest pods that seem to appear in the roster of people who are attending this workshop and who, sort of indicated what kind of substantive research interests they have. The way we organize tomorrow is you can work on your own data or a model data set that we distribute through a Google Drive folder that there's a link to, I think, in the email that you got. So you'll have data that you can work with. You'll have R script if you want to do it in R. Similar things can be done in just about any data analysis package. It is completely feasible for simple situations to actually analyze RNA data in Excel. So there's no version of statistics, no version of software that is absolutely required to get into this. But since most people seem to be converging around R, we've sort of used that as our default, sort of model syntax. In tomorrow's session, we'll probably have folks work together if they don't have data of their own, in part because for people without much, sort of, if you will, kind of tactile contact with data, it's often kind of in sort of puzzling how you get started, or even how you get data read in, or what you're going to do with this error message that says there's too much data for the piece of software that you're working with, or something like that. So if you guys want, you're perfectly welcome to assemble into groups. You can assemble into the groups based on these substantive, similarities that we saw in the attendee list. You can self-aggregate into groups based on, we're all from the University of Michigan, we know one another, or we're all involved in the same study or something like that. So just because we have a list of people who are interested in, let's say, early adversity and its impact on lifespan aging, doesn't mean you guys all have to go sit at the same table and work together. It's just an idea about what you can do. You don't, there's lots of us who are introverted, antisocial people. We have a good idea what we're doing. We really don't feel like I want to get together in a big group. That's also perfectly fine. So we're going to do that kind of stuff basically with our hands tomorrow. And then mid-morning tomorrow, we're going to have like a report out on how that went, in part because what we've seen is that, most of us are going to run into more or less the same challenges. And so the more we can kind of make it clear that the challenges you're running into, they're not you, there's nothing wrong with you, that in fact, these are kind of generic challenges. And that other people, some of them have actually worked all this kind of stuff out. Some of these challenges are technical, some of them are conceptual. So think of tomorrow not so much as like just a technical practice with data, as a sort of a collective group learning session, really more than anything else. And so tomorrow, it's definitely going to be good to take lots of time and talk about your experiences. So we've made tomorrow have essentially no didactic information, and sort of mostly sharing or sitting there kind of at work at your own table. Does that all make sense? Any questions about the format of the day? Anything I missed, and anything not so clear? So the first thing I'm going to do is talk about, actually, can we get the lights down in the front of the room so it's easier to see yeah, it's the panel on the left there. So I think it's labeled. Yeah. So Zoom needs screen shared. That's nice. Zoom is, for some reason, oh, sorry, Zoom is not. Wow, Zoom really does not like presenter view. Anyway, so first question, why are we here? Why are we bothering with RNA? What is the point of this whole exercise? So in general, I'm going to assume that most of the people in this room are going to answer that question in terms of learning how something in the social or the psychological or the economic or cultural or political world, all of this life outside us or in our minds or whatever, how that stuff actually turns into something that is biologically us. So in most cases, this is going to be things like health outcomes, but for some of you, it's going to be like developmental processes, like how your immune system develops or how your brain develops or your, sort of the development of various capacities or capabilities. So broadly speaking, we're going to be talking about this flow of information from essentially a social world outside the body into the tissue of the bodies with some kind of phenotype of interest like health or development, behavior, that kind of stuff. And it's helpful to unpack those outcomes into something in terms of how they actually come about in the body. So what aspect of physiology helps you grow, a healthier lifespan as opposed to a greater risk of disease? What confers the capacity for some behavioral outcome or social outcome or economic outcome of interest? So you can sort of think of reverse engineering the outcomes we care about into biological processes. So for instance, in the context of health, we might think, hey, one of the things we're worried about is, are people dying? You can reverse engineer that into questions about, are they getting cardiovascular disease? Are they getting cancer? From that, you can reverse engineer into like, are they showing higher levels of inflammatory biology that would, let's say, make a respiratory virus infection into suddenly a lethal pneumonia or something like that? So that kind of reverse engineering of the phenotypes that we ultimately care about is essentially what is motivating this focus on RNA. So that reverse engineering of the physiology and behavior usually ends up being tantamount to a question about how does the behavior of cells in a body change as a function of, let's say, these social environmental conditions. So most of those cellular changes are actually mediated by changes in the production or maintenance of proteins, either the production or maintenance of proteins that create a new cell that wasn't there before, or that either maintain a cell in its current state or allow the cell to adapt to some kind of new world that it's living in. So most of you can think of them as sort of biologically or health or developmentally consequential changes in how cells work are mediated by proteins, because proteins are essentially the muscles and machinery of cells. Cells have all kinds of other stuff going on inside them chemically and molecularly, but in terms of actually getting stuff done, the bulk of the answer to the question about why can this cell do this and that cell can't is essentially a question about why does this cell have the proteins that allow it to do this and that cell doesn't. So now we can reverse engineer this question about death into things like cardiovascular disease and into like lower order physiological processes that might promote cardiovascular disease. Now we can get very concrete and say, actually, we don't mean inflammation in general. We mean literally the production of interleukin-1 beta protein, or the production of, sort of COX-2 enzymes, or the production of interleukin-6 or things like that. So these become sort of a, if you will, kind of like a tangible molecular reification of the constructs that we're ultimately interested in. But there's this next question then, which is like, okay, well, why does this cell have these proteins and that cell doesn't? Why is it that, this cell is adapting well to a world of stress or threat or uncertainty and that cell isn't? And as a consequence, we're seeing these sort of changes in the flow of biology that become the phenotypes we care about. So again, there are lots of different biological processes involved in sort of either maintaining or changing the complement of proteins in a cell or a body. But the dominant influence on that sort of profile of proteins that are being expressed or active in a particular cell at a given time is the messenger RNA that guides or serves as the template for the production of proteins. So most of us are used to thinking, like that our bodies are stable biological entities that are sort of like, we live in the world but are separate from it. In other words, we ourselves are like a stable kind of biological thing that goes around from one environment to another. But actually, it turns out when you look at the minute biological level, we are all dying all the time. All the time, our cells are getting broken, damaged, dead, and we are constantly reinventing ourselves by replacing those lost cells, or by replacing the lost or damaged proteins, or by producing new proteins to deal with some kind of new environment that we live in. So at the micro level, at the cellular level, we're much more sort of dynamic, much more fluid than we typically experience our bodies as. So this process of regenerating protein or adaptively changing our protein structure is fundamentally guided by the human genome. And that's kind of the magic of what it is. It's not so much that the DNA by itself is that incredible as, one, it's kind of an efficient way of packaging a menu of options for making proteins. But two, there's this sort of millions of years of acquired wisdom baked into the wiring diagram that turns on and turns off proteins. In other words, you can think of it as a very highly evolved, relatively wise system about how to change your proteins in response to changes in the world around you or changes in how you're experiencing that world. So all of that information ends up being codified at the level of DNA. So the chief determinant of whether you've got the proteins you need or you've got the proteins you don't want in your cells has to do with the production of the messenger RNA that guides the production of proteins. And those messenger RNA is essentially, if you remember from high school, college biology, that this is kind of the template for which proteins are going to be produced in a particular cell at a given time. And that messenger RNA is what's being read out of the human genome. So human genome, big stretches of DNA, small little segments of it that are eligible for transcription from their original DNA form into an RNA form. And then that RNA form gets translated into a sequence of amino acids that constitutes the protein that's going to confer the good things that you can do or your risk of disease or that kind of stuff. So this is the real reason we worry about RNA, is that it is, in some sense, the sort of, not just a sort of in a flow of information sense, a root cause for the changes in protein biology that we care about in terms of many of the phenotypes that we're interested in, but that it is a sort of an unusually, not just sort of close to the root, but it turns out it's the first environmentally variable thing in this arc off of DNA. In general, your DNA is not going to change in response to your environment. Your RNA will change quite dramatically in response to your environment. Now, there are some minor exceptions. If we drink a lot of benzene, our DNA will change. It will get nicked and dinged and that kind of stuff. But and there are certain cells that actually, just for the fun of it, scramble up their DNA because this is an efficient way to, let's say, try out different receptors for microbes or something like that. But by and large, DNA is constant in your body from the date of your conception, essentially, and will be more or less the same when you die, at least in terms of DNA sequences, as the day you were conceived. RNA is all over the place in response to what's going on around you, and in this case, the you that counts is actually the cell harboring the DNA. So RNA is also going to be very different in this cell versus that cell, and in fact, that is the definition of what a cell is. It's the subset of DNA, potential molecules that are actually realized within this cell. The cell is a neuron precisely because of the subset of RNAs that are expressed. And if it took the same DNA genome and expressed a different subset of messenger RNAs, it would be an immune cell or a muscle cell or a liver cell or something like that. So this kind of heterogeneity is, sort of, if you can think of this as like the chief challenge and the chief opportunity in RNA research, unlike DNA research, where, as many of you are aware, you could take salivary DNA samples and get more or less the same information as you get from, let's say, a white blood cell or a muscle DNA sample, if you're just interested in nucleotide sequence. And then many of you have kind of bumped into the world of epigenetics, where there's some inconsistencies in that at the level of modifications of DNA, like methylation. Well, RNA is the extreme Wild West version of this, where it's totally different from cell to cell and potentially quite different from time to time. So this is, again, becomes a data analysis challenge, but it's also the opportunity, because now you're starting to see life happening in response to these social and environmental conditions. So how is it then concretely that the genome actually gets played on by these kinds of external experiences or external stimuli? That happens concretely at the level of DNA when essentially a molecular switch called a transcription factor binds onto DNA in some position that essentially works as like a flag that says, okay, for whatever, and we'll talk about we'll unpack this next statement of like, for whatever reason in a moment, but at the moment, just suffice it to say, what actually turns on the transcription of RNA is the binding of a DNA, a protein called a transcription factor generically onto the DNA, usually a little bit upstream of where the coding region of the DNA, the part that's going to be transcribed into RNA is present. So this transcription factor you can think of as like a flag that basically says, hey, come transcribe this part of the human genome. So there's literally like a free-floating DNA copying factory called RNA polymerase that will see these flags and say, oh, okay, this apparently is a section that needs to be transcribed. So I'm going to land on the DNA here and start copying this off into messenger RNA until I reach some kind of stop signal, basically. So the key on-off switch for RNA is the presence of these transcription factors in what we call the regulatory region of a gene, or sometimes we call it the promoter or the proximal promoter. So the transcription factor is literally the thing that determines whether this stretch of DNA is going to be transcribed into RNA and then become protein and change cellular function and behavior. So stepping one click back, most of these questions, as we reverse engineer them, then become questions about transcription factors. And the nice thing about the transcription factors is they're a good meeting point between this flow of information out off of the genome and the flow of information into the proximity of the genome from the social environment. So how does that happen? Well, basically, some transcription factors are regulated by the internal logic of the cell, but many transcription factors are hooked up to receptors on the surface of the cell. There's biochemical signaling cascades that are triggered by receptors when, let's say, they bind to some kind of a molecule, like, let's say, to use a concrete example, let's say I've got an adrenergic receptor on the surface of a white blood cell. That adrenergic receptor can detect when fight or flight stress molecules are produced, neurotransmitters like epinephrine or norepinephrine. So this adrenergic receptor will say, hey, I've just bound some norepinephrine. I'm going to kick off this biochemical cascade, and that is going to increase, or sometimes the system is wired to decrease, the activity of often more than one downstream transcription factor. So perhaps multiple transcription factors will be activated, and perhaps additionally some of them will be suppressed when this molecule outside the cell gets detected by the receptor on the surface of a cell, or sometimes inside the cytoplasm of the cell. But this then becomes essentially sort of the causal flow that drives the variation of transcription factor activity. And so when we're talking about social environmental influences, there's really at least two broad ways that they can become signals that interact with these receptors. One of them is in our physical and built environment. There's all kinds of physicochemical and microbial stimuli that end up being detected by receptors in cells. So the social world in part changes the world that we live in, the amount of, let's say, tiny particles we breathe into our lung, or benzene in the drinking water, or sort of infectious viruses or things like that, all of which end up being detected by receptors in cells that then kick off these signaling cascades that result in transcription factor activation. So that is literally what happens when you get let's say, a viral infection. Is you've got, viral DNA or RNA detectors that go, hey, there's viral stuff here, kick off an immune response, and that's driven by these particular transcription factors. There's a second major wing of social environmental flow that comes from our experience of the world. And this is really closer to the example I gave earlier with the norepinephrine. It's like, if we are, even if the external world isn't hitting us with different microbes or different chemicals, even if it's simply our experience of the world that varies, that too becomes some kind of biochemical representation within the body. So in the scenario I was talking about earlier, if you're feeling threatened or even just insecure for an extended period of time, you will have more of this fight or flight signaling going on inside your body that will produce more things like epinephrine and norepinephrine in your blood and other tissues. And those signals then will get detected by these receptors and kick off these signaling processes within the cell, eventuating in changes in transcription factor biology. So this is the ultimate reason we worry about RNA, is because one, it is, if you will, the first sort of environmentally contingent, event after this relatively static DNA that's going to reflect how the environment is operating. Another reason, though, that we like RNA is that we are really, really good at measuring RNA and DNA, way better than we are at measuring most other kinds of chemicals or proteins or other substances in the body. There's a lucky quirk of the chemistry of DNA and RNA that essentially makes it much easier to, sort of detect one molecule. Like it's a cakewalk to detect one molecule of RNA in, 10 billion copies of RNA. If I know what I'm looking for, I can find it. We have great chemistry for this in ways that we just don't have for things like proteins or many chemicals. So that, combined with the fact that there's a non-trivial number of genes, about 20,000 genes encoding about 50,000 different gene products, different alternative versions of a gene transcript. But actually, interestingly enough, because we're so good at this kind of base pair complementation chemistry that we see in DNA and RNA, it's actually not particularly difficult at this point in our amazing technical evolution to count the number of molecules from every single gene in the human genome. In fact, that's what we're mostly going to be doing with the data sets we have. We're nowhere near being able to do that with any kind of chemical or any kind of protein, but counting molecules at the level of DNA and RNA is really easy. So the advantage of RNA is that it's the environmentally malleable version of this. The advantage of DNA is that it's the ancestrally, if you will, acquired wisdom that, sort of interacts with the social world to produce this net vector of RNA that ends up being this immediate determinant of health. So that's why we put up with it. Sorry for the tedious, sort of introduction there, but many of the complaints we have is like, I'm a behavioral scientist, there's all these molecules, which one should I do and why, right? So having a sense of how it all fits together does two things. One, it gives you some sense of what you're doing and why, but also too, it helps you realize that there's not one answer to the question of what you should be doing. And in fact, this is really a story about how these different molecular and environmental streams of influence come together and interact with one another. It's not like one is right, the other is wrong. They're different pieces of this ultimate puzzle about why the social world becomes, let's say, the health or behavioral phenotypes that we care about. Okay, so from this kind of rarefied conceptual world, now we're going to do an incredible descent down to pragmatic, details of all of this that are, once you have that conceptual roadmap laid out, actually a little bit easier to understand. One of the questions that is particularly significant in RNA in a way that is not so much the case for DNA, although intermediately so for things like methylation, is where should we be collecting the sample that we're interested in? So for most biologists, and I have to confess, I was once upon a time trained as a behavioral scientist, but they mostly threw me out. And so the people that took me in were biologists. So I'm going to be mostly talking about what we're doing kind of, see, how should I put it? Through the lens of a sort of social or behaviorally science biased biologist, essentially. So, there's all kinds of reasons to maybe don't necessarily get preoccupied by the things that I'm preoccupied by. I'll try and decenter and mostly tell you stuff that I think you would be interested in. But one of the things that we in biology care a lot about is the locale and timing of tissue sampling. And that's because we don't think that any of this stuff is the same all the time or in every tissue, right? So this kind of default assumption that there's a genome and it's operating, like this makes no sense to most of us in biology. It's like a genome in what, where, doing what, with respect to what outcome do you care about? Like we understand there's DNA there, but as we just talked about, that's not enough to understand. We used to give lectures where we would take, whatever, 10 million copies of the human genome and put them in a little tube on the podium. And then at the end of the presentation, we'd be like, look what the human genome by itself has done. And it hadn't done anything, right? It just sits there floating around in water. And that's because inherently it needs what we would call the spark of life. It needs other stuff to interact with it for it to do the magic that it does. So where and when and how we should sample actually looms pretty large in the RNA world. DNA, mostly the same in every cell, subject to the exceptions that I talked about earlier. RNA, by default, we assume in biology is different in every cell and tissue. Maybe, tissues which are aggregates of broadly similar cells, maybe they've got fairly well correlated RNAs, but we still sort of believe that, even at the cellular level, there's a huge amount of wax and wane in this sort of RNA production process, both in response to the environment and in response to local signals generated by other cells of the same type. So there's about, you can change the degree of stringency in the definition, but to a first approximation, you can think of there as being, broadly speaking, about 200 different types of cells in the human body. And so picking which of those cells we look at, it looms extremely large for the RNA world because each of those cells is going to be expressing very different complements of RNA, which is the basis for their differences in cellular identity. And this is like a really old time plot, but the visual bars should tell you that, for every single protein in the human body, you get variations on anywhere from like 1,000-fold to 1,000,000-fold in the abundance of this particular protein across cells. For many cells, the protein is literally like zero expressed. It's not there. For other cells, there's, hundreds of copies or thousands of copies or sometimes millions of copies of this protein there. So choosing which cells you look at makes a big difference. Even in tissues like blood, which we think of as fairly homogeneous, it turns out comprised of lots of different types of cells. The dominant cell that you see there, those red blood cells, they don't even have DNA. They've already ejected their DNA and most of their RNA, and they're kind of like, leftover ghost cells or zombies or something like that. So a fair amount of variability in which RNAs are expressed, even within different cells within the same tissue, like blood. And the complication that comes up for RNA there is that the composition, the relative prevalence of these different types of cells can change actually quite dramatically in response to environmental conditions. So I'll show you one example that plagued us. I was basically, when I was thrown out of behavioral science, it was immunologists that took me in. So I'll be showing you some immunology examples, but they for years have been focusing on the effects of stress on the immune system. So here we were just looking at what happens when you activate the fight or flight stress response. We had some poor undergraduate run up and down 14 flights of stairs to get her autonomic nervous system going. Took some blood before and after, and she was really interested in transcription factor activity. But to understand that, we wanted to make sure that we were measuring the different types of white blood cells that were present in her blood. And you can see that, on the left here, easier to do if I use my pointer, but in this left panel, what you're looking at on the left-hand side down here with SNS activity negative is essentially Vanessa when she was just sitting there all chilled out, and then Vanessa after she had run up and down 14 flights of stairs. She did this a number of times, so we had a pretty good sense of how reliable these effects are. So what you can see is there's pretty big changes in the absolute abundance of various types of white blood cells. You're getting, so it looks like about at least a 2 to a 3-fold increase in the absolute number of monocytes and the absolute number of lymphocytes. What exactly those things are doesn't matter so much as they do different things as part of an immune response. So that matters. And, they express very different RNA profiles. So that also influences what you're going to get out of one of those RNA profiles. But notice the granulocytes, which are actually start off being the most abundant type of white blood cell in circulation. Once you've stressed her, you actually have granulocytes now being the number 2 most prevalent white blood cell, surpassed now by lymphocytes. So as a consequence of that, essentially the relative ratio of these cells changes over time. In this case, within about 7 minutes, as Vanessa ran up and down these flights of stairs. So this kind of biological variability isn't just a slowly evolving response to, again, benzene in the drinking water. Like, very rapid changes can occur in many of our tissues, it turns out, as a function of thinking or living or running or being afraid, that kind of stuff. Even when we look within the lymphocyte pool, there's different types like T cells and natural killer cells and monocytes and B cells. What you can see is many of those types of cells like T cells and B cells and monocytes actually slightly trend down as a percentage of total lymphocytes. But there's one subset called natural killer cells that ramps up dramatically as a function of this. So these cells, again, have different RNA profiles. So as a consequence, your white blood cell pool, every RNA that is more abundant in natural killer cells will look like it has been upregulated if you take a blood sample after you've been stressed versus before you were stressed. So we in biology worry about this stuff a ton. Like, we don't even believe that sampling from the same cell type is enough. You need to normalize the conditions under which you sample ideally. So this idea, this mindset that RNA is local and that it is it's both physically local to tissue types, and local in time or circumstances is kind of one of the overriding differences from what we do in genomics. So RNA can change even within tissues, definitely over time, and with things like disease. So on some level, that's a confounder or a hassle, but on another level, as I just described, that's the opportunity that lies before us, right? We can use these changes in the RNA to understand the development and production of disease or health or all the other kinds of behavioral things that we're interested in. So this is part of the reason that you'll see a lot of attention to cell types in the world of RNA analysis, and that's becoming now actually more prevalent in the world of, let's say, DNA methylation, as well as this greater appreciation of the significance of those effects. So, one of the things that we do is say, okay, first, I need to decide where I'm going to sample based on what kind of outcome I care about. There's no point in sampling from brain tissue if I'm trying to understand heart attacks, right? It's just not going to work because they're completely different physical tissue compartments, different neighbors, different stimuli. There's a blood-brain barrier between these things, all kinds of reasons not to try and use one type of cell as a proxy for another. So in the RNA world, the default assumption is you can't just take blood cells and assume that the same transcriptional dynamics are taking place in the brain or in muscles or in reproductive tissues or in the gut or stuff like that. If you want to understand the gut, you need to physically sample the gut. General people will not accept blood cell RNAs as signs of what's going on in the gut, but they will accept the idea that some of the cells in the blood will eventually travel to the gut. So there's a version of this where even if you can't get people to give you brain tissue, all you can get is white blood cell RNA. We now know white blood cells go pretty much everywhere in the body, at least under some circumstances. And so they are broadly relevant. But what you don't want to do is start with this assumption that this is the same RNA genome operating in white blood cells that is operating in, let's say, brain tissues or gut or stuff like that. All right, so what that means in general is that we're going to want to tissue sample from the tissues that actually matter for the phenotype that you care about. If you want heart disease, sample from the heart or from the white blood cells that will interact with an atherosclerotic plaque. If you want to understand the brain, sample from the brain. But that makes life kind of hard because it's hard to get people to give you their brain tissue. So, this constrains to some extent the kinds of questions that we can feasibly ask. So with that in mind, just be aware that as you're making, claims about implications, let's say from the blood cell RNA profiles in HRS or AdHealth or MIDAS or something like that, you acquaint yourself a little bit with what's known about how the white blood cells that are supplying these RNA profiles might physically participate in the phenotypes that you care about. There's another set of questions about how you should even get your tissue samples in the first place. Traditionally, what we've done is directly sampled essentially surgically, right? I mean, we'll just physically go into tissue and invasively pull out chunks of it, like in venipuncture or in surgery or something like that. So, there are some great advantages of that, but the downside is it's not a very field-friendly approach. So historically, this stuff has been confined to laboratory environments. And, basically that eradicates a lot of complications that come up with other modes of sampling. But recently, it's become much more feasible to sample RNA from other environments. So some of the innovations that we've seen involve just reducing some of the hassles that, sort of accrue in the laboratory collection environment. So one of them is that, as I mentioned, RNA is constantly changing over time. That includes when you draw blood out of a person. So if you take blood out of a person and you leave it, RNA will start changing. That effect is not very large for the first maybe 5 to 90 minutes or something like that. But slowly over time, that RNA is going to change. And in fact, when it changes dramatically is about an hour to an hour and a half after blood draw, when a bunch of those red blood cells have, they basically start lysing, they blow up, they kind of explode, and that creates a bunch of tissue debris within the blood sample. That tissue debris is one of the major things that the immune system reads out as a prompt for activation. So the immune system goes around patrolling for lots of things, including damaged human cells. So when the red blood cells blow up, that's read out by the white blood cells in this tube of blood as, oh, there's a bunch of tissue damage around here. And they start in on inflammatory reactions and early stage immune responses. So that's the reason we don't like to draw blood and then just kind of leave it around. That really makes things difficult to interpret. So one of the strategies that has dealt with this kind of time issue is like, once upon a time, we were literally, if we wanted interpretable RNA data, we had to be within a situation where we could get the blood cell, processed within about 15 to 30 minutes of its collection out of the vein. One of the things that has changed the world of RNA analysis a lot is the production of RNA stabilization systems. So the most commonly used one is this PAXgene RNA tube, which has a little sort of aliquot of about 1.5 mils, kind of like a pinky size volume of fluid in the bottom of it, that when the blood goes in there, it blows up the red blood cells, it blows up the white blood cells, and it wraps up the RNA in a chemical blanket to keep it from being degraded. So you can draw blood into a PAXgene RNA tube, leave it at room temperature for a week, and it's still fine. I would recommend following the manufacturer's instructions and putting it in a freezer within 3 days, but honestly, this works pretty well, even when you kind of push the limits of what it does. So this has made a big difference because now essentially RNA analyses are mailable or drivable. You don't have to bring people into a laboratory to get an RNA profile that looks very similar to its actual native original biology. You can sort of suspend the laws of time and space or something like that a little bit. It is also possible to get RNA out of saliva. There's RNA in there. We generally don't recommend it because the RNA in saliva is often degraded. And that's because that's saliva's job, it turns out, is to like degrade weird stuff that's going into your body. So your saliva is filled with RNA degrading enzymes called RNases. Your saliva is also filled with a bunch of sort of cheek cell gland RNA and white blood cell RNA. And there's a huge amount of variability in the relative ratios of those things. And then all of us have a fair amount of bacteria, but some of us have a ton of bacteria. So this is like really sort of a big complication in salivary RNA. And unfortunately, the amount of oral hygiene related bacteria correlates very strongly with some of the substantive variables that we actually care about, like developmental status or socioeconomic status or that kind of thing. So while you can get RNA out of saliva and it seems very convenient, it is basically very challenging to interpret. So I wouldn't necessarily recommend doing that unless you are interested in something where, you know, essentially the inside of the mouth is your chief biological, you know, or substantive interest. So don't use it as a proxy for the immune system in general. But if you're interested in, you know, literally like why do people's teeth fall out as your substantive question, then it's probably worth doing. Another thing that has even more greatly, you know, sort of expanded the scope of RNA work is the ability to retrieve RNA from dried blood spots. Dried blood spots, as you guys know, are great because you don't need venipuncture and phlebotomy to get them. You can get people to collect their own blood samples out in the field, and then basically, you know, you can take weeks or months or sometimes even years to get those samples out of the field and into some kind of centralized laboratory environment for analysis. So this has really significantly expanded the scope of RNA out of the world of what you could think of as like clinical research, and even out of the world that's kind of relatively typical of this room for kind of like, you know, population health and social and behavioral research in developed nations. And now you can do this stuff, you know, basically cultural anthropology style. You can get these samples anywhere. Interestingly enough, what happens is when the blood dries out, that doesn't actually damage RNA very much, but it deactivates the enzymes that would otherwise eat your RNA. So this whole process of drying, which sounds terrible, actually turns out to be great in terms of preserving RNA and DNA. So it's got its downsides. There's relatively little blood in a blood spot compared to like a big PAX gene tube. So there's more noise from the law of large numbers, essentially, because this is a smaller number of cells. So essentially sampling variability is bigger. And there's a certain amount of breakage, but the RNA assays that we have now are actually pretty tolerant of RNA breakage. So dried blood spots are definitely feasible. They're noisier. They're not, you know, what we recommend is the first approach for anything. But if the difference, if your question is, you know, do it, you know, with venipuncture or don't do it at all, you know, there's now another option, which is you can do it with this noisier sampling situation. Most of the time when we run the numbers, you need about a 20 or 30% bump to sample size to offset the, you know, signal to noise cost in doing dried blood spots. Many of you also may not be aware of this, but there is a new generation of dried blood spot samplers available where you can literally mail people a box. It has everything that they need. They collect their own blood samples and then they put it back in another mailer and send it back to you. So this made things very much easier during the kind of the COVID social distancing era. Yeah, you had a question? Well, so PBMC, peripheral blood mononuclear cells, are the predominant, you know, sort of cell sample that laboratory, you know, biologists, particularly immunologists, have historically collected. So it's the most common source of, let's say, DNA samples or RNA samples that would be validated as predicting a disease process. So you can think of them, you know, they're sort of the default gold standard. They're sometimes not the actual perfect gold standard because, you know, often they're somewhat remote from some of the things people are trying to understand, like heart attacks or neurodegeneration. But there's a huge kind of, you can think of it as repository of information about RNA profiles in this particular type of sample and how they predict various disease outcomes. So validated against PBMC basically means this sampling strategy, you know, gives you something approximating, and it varies a little bit across sampling modalities, but this gives you something approximately as predictive of disease outcomes as PBMC would. So it's, you know, it's one of these things where that's, for instance, what makes the salivary RNA sampling not that great, is that it really is noticeably different tissue from PBMC, this supposed gold standard, whereas PAX genes and DBSs are closer, even though they have their own complications. There's red blood cells that are in the PAX gene and the BBS samples that there wouldn't be, let's say, in the PBMC samples. Yeah.
Oh, I think I understand red blood spot. Want to make sure that it's dry. That means like maybe bone or this. There are some protocols that maybe are a little good for drying and a little bit for other.
It would not be ideal. They'll still dry out surprisingly well. Most of the drying that takes place in the cards is actually by the card wicking off the fluid. So, you know, that said, it's definitely better to dry it out as much as you can, as fast as you can. So yes, to your initial intuition, but don't panic, you know, in the pragmatic world where people don't always do what they say. The consequences of it being a little bit slower to dry out don't seem to be, you know, catastrophic. So it's not the kind of thing that I would think would, you know, drive you to be hesitant about using that modality. It's more the kind of thing that would drive you to make a swanky little YouTube clips where you remind people to, you know, actually dry things out. That's part of what makes these new generation samplers a little bit better too, is they you know, they both have dry out accelerators to try and, you know, sort of make sure that stuff gets done, but they're also optimized to do a fair amount of drying, even if somebody decides they're not going to pull the plastic tab that, you know, activates the dry out accelerator. The magic for this stuff mostly is the first maybe 30 minutes or so. If it's dry within 30 minutes, RNA is sluggish, so it's going to take a while for the RNA to start deviating based upon the wetness, essentially. So as long as you can get it dried out within 30 minutes to, you know, a reasonably, it doesn't have to be perfectly, you know, water-free by that point. So in the empirical world, it tends to work reasonably well, even though it's definitely a reasonable concern. Yeah.
So,
We like a whole circle, a really good circle. And in fact, we really like two of these things. You know, there's no magical line in the sand. So you can work with anything, but the smaller drop is going to yield less RNA. So you're going to be more victimized by, you know, the law of small numbers, basically, which law is like crazier stuff happens. And, you know, you'll end up with metrics down the line that say, you know, this is quantitatively somewhat less reliable than, let's say, its neighbor that was a big spot. So usually the best thing to do, I mean, the problem is that you can't get everybody to produce a big spot, right? Some people just won't do it because they're not into it. Other people won't do it because they can't produce that much blood. So, you know, in the world that we live in, you just work with what you've got. And, you know, usually we don't throw things out based on that unless the spots are really small. We give it some kind of a quality score that then becomes a weight in your downstream data analysis. And you just say, you know, I'm not going to believe stuff from this quite as strong as I believe stuff from a big spot, but you know, neither is it nothing. And I don't really want to be throwing out small spots, especially if they might be correlated with social or behavioral characteristics that might be of interest. There's a question. Yeah. Oh, yes, I can paraphrase the question before I answer it. Yes, thank you. I'll get all of you, so go ahead.
if you are collecting whole blood, is there a way to maintain is it okay with the way
Yeah, so the question is, how do you ensure the integrity of, you know, wet blood samples, and, you know, should you be really concerned if it takes you more than 15 minutes to get it to minus 80 degrees or something like that? And the answer is no, in general, you shouldn't. The primary answer to how to maintain the integrity is actually these PAX gene RNA tubes. So those things freeze the RNA profile the moment the blood hits the tube. So they, that after that, there's almost no risk of degradation unless you leave the PAX gene tube in the sun for months or something like that. Like you really have to work to degrade it there. So that's the primary solution. When you can't do that, which many immunologists don't like PAX gene tubes because they want to isolate white blood cells or something like that. That's great, but the RNA profile is drifting over time in every white blood cell that you are messing with while you're messing with it. So the biggest concern is actually highfalutin immunologists who spend 2 or 3 hours sorting out tiny little subsets of white blood cells, seemingly without awareness that those white blood cells are changing their stripes all of that time. Now, the way they manage that is by trying to keep the cells cold. So that's the second answer to your question. If you want to do some long drawn out processing, then usually you pay attention to the temperature of the cells. And the idea is that if you slow the cells down, which is what happens when you cool them, that they will produce RNA deviations at a lower speed, basically. So there's another question here. Yeah. Yeah, so the question is, what is the ideal time frame for drying on dried blood spots? So the answer is it is a little bit variable depending upon the circumstances. So for instance, we were doing this crazy study of child soldiers in Nepal or something like that. You could leave that thing on the counter for weeks and it would not dry out. It's not going to dry out. It's Nepal. It's like ambient humidity is, I don't know, like 85%. Something like this. It's crazy. So the solution there is actually to get it dried out enough that it doesn't smear when you sort of close it. And then as quickly as possible, you get it in a Ziploc bag with a desiccant pack. And there it's got its own little atmosphere that is actually much drier than the ambient atmosphere. So for most dry blood spot protocols, the way you manage this is you do a modest amount of drying, like, whatever, 15, 30 minutes, at ambient, and then basically you close it up and you give it its own little atmosphere with a desiccant pack. And that's, many of these systems that are, sort of built like that, the old Tasso M20, have a dry out accelerators basically to help with this kind of thing, and desiccant packs and their own little Ziploc stuff. So that's part of the reason lots of people are working with that now. Is like it just comes with everything you need. You don't have to create the pack. The pack comes as a box. You can mail the box. People send the thing back. It's quite a cakewalk. They're expensive, so there's something to be said for if your undergraduates are bored and you want them to put boxes together, you can probably save money. But the upshot is, as long as you get them into their own drying ecosystem within maybe 30 minutes to an hour or something like that, you're generally gonna be fine. There won't be too much trouble. There was another, okay, yeah. You can put them in the fridge. That is better than not putting them, anywhere. So the question is, she hasn't heard me saying anything about the fridge. Putting them in the fridge is great because the fridge actually drops humidity a fair amount. It's not perfect, but it definitely dries things out, as you know from putting your lettuce in the refrigerator, right? Eventually it will suck all of the moisture out of your fruits and vegetables. It will do the same thing in there. The other advantage of being in the refrigerator is that the temperature is lower, and so all of these enzymes that would be eating the RNA will operate with less efficiency, so you'll get less degradation. So fridge is better than room temperature, but if you can get them frozen, that's even better. The incremental value of freezing, it's there, but on the other hand, the incremental cost of freezing is non-trivial. So usually what we try and do is the best that's easily feasible in the ecosystem. Like in the Nepal study, people would walk samples or ride samples into local laboratories, and then they put them in a refrigerator there because you couldn't get a -80 freezer out into the fields in Nepal. And then those guys would monthly send in their batches to some place in Kathmandu or something like that, where they actually did have a -80 freezer. So it's basically just optimize what's feasible. But the whole point of DPS is making things feasible. So there should never be a situation where you say, I'm not going to do it because I don't have ideal temperature control. Is So usually they're in their own little Ziploc bag, right? There's a dried blood spot. It's in a Ziploc bag with a desiccant. That at that point, it's mostly, that's 80 or 90% of the preservation. If you want to freeze them further, especially if you're going to acquire over long periods of time, then it's not a bad idea to get them into -70 if you can. Again, monthly batches going in is great. The point of -70 is to keep it stable for months to years or something like that. Not so much like you need to get it there within 15 minutes. So that would be the ideal way to save it. That said, surprisingly, stuff that's been at room temperature for years still performs pretty well for many assays. It quantitatively degrades over time, but there's dried blood spot studies where they have decade-old heel sticks from infants or something like that, and they validate them. And they work well, but they're noisier than something that had been at -80. So it's all about your kind of trade-off of feasibility. Okay, any other questions at the moment about that? Great. All right. Next thing that we'll go on to is, what happens to these samples once we've actually got them in hand? We've acquired our samples. Now we need to get RNA out of them and make sure that RNA is actually useful for downstream assay purposes. I'll tell you, these days, the downstream assays have become very good. So almost everything is useful for downstream assays. This didn't used to be the case. And there are still some assays that are kind of high maintenance and brittle, but most assay systems have become much, much more tolerant of partially damaged or suboptimal RNA. So we'll talk a little bit more about that later, but basically you've got your tissue sample. Once upon a time, we had this great, sort of montage of all of the various steps where like undergraduates were dutifully, sort of isolating like, sort of organic chemistry layers and stuff. But honestly, everything is done by gray boxes now. The robots have, taken over the world. So essentially what happens is blood samples get loaded into these robots, which are really centrifuges with cranes that move, sort of little dollops of organic chemistry fluid around in various sequences that will eventually hold up the RNA in a little matrix of RNA binding chemicals, basically, while letting everything else pass through. They use the centrifuge to drag everything else. So all of the lipids and all of the chemicals and other stuff in the cell gets pulled through this matrix, but the matrix loves RNA, so it holds on to the RNA as it goes through. And then when you drop some water in there, magically the matrix lets go of the RNA. So you can pull everything else out and then basically drop some water on top of these things and centrifuge out just the RNA. So now you have RNA in a little, I don't know, I usually bring one of these things. I forgot to do it this time, but a little 1.5 mil, microcentrifuge tube or something like that. But it's literally, it just looks like water in about 10 microliters of fluid. You can't see the RNA. It's not like a pellet or a chunk or anything particularly colored. The first thing we're going to do with that, now that said, when the RNA is extracted, it is extremely vulnerable to degradation. So this part is where the stakes suddenly get high in terms of experience processing. When RNA is in tubes, especially taxgene tubes or dried blood spots, it's actually surprisingly resistant to degradation. So we beg people not to extract their RNA and then send it to us, because if FedEx decides to park that in Tennessee for a weekend while there's a strike or something like that, that RNA by itself is going to be gone. RNA, what we call naked RNA, is incredibly vulnerable to degradation. So usually you want to keep your samples in their original native form, where the RNA is surprisingly more stable than it would be if separated from everything else, and then send it to us, and we'll do the RNA extraction. When I say to us, I mean people in the laboratory world. You don't have to send it literally to me, but send it to the folks that are running the robots, and essentially, try and get all of that done in the same physical environment where you're actually having the assays perform, ideally within the same few days. So if there's a relatively minimal period of time where the RNA is in this kind of naked, unprotected form. As long as the RNA is frozen, it will be generally fine. But you never want it to actually come to room temperature. So people who do RNA stuff, one, you'll notice they look like, sort of like COVID, concerned people long before COVID concern was a thing, right? They were always wearing masks. Why? It turns out your breath has RNase enzymes in it. So they don't want to be breathing, RNA degrading enzymes into the samples. They were wearing 2 layers of gloves because they want to be able to change their gloves every time they pull their gloves out of the biosafety cabinet. They do everything inside these biosafety cabinets with these air curtains to prevent microbes from getting in there. There's a bunch of other stuff related to like not touching the top of the tubes or stuff like that. So there's this whole world of basically RNA safe practices that suggest that, I mean, you can do this stuff yourself, it generally works, but you're definitely safer, and especially if the stakes are high, sending it to somebody who has experience doing, RNA handling, just because you're much less likely to run into this kind of degradation, at least after the extraction of the RNA. There is some degradation that can happen for upstream reasons, but most of it that we've seen happens downstream. And what you never want to see, like we see this all the time, it's very disturbing, but like, you'll see like thousands of RNA samples that look great, and then you'll see 96 that look terrible. And you're like, no, that's not how the natural world breaks down. 96 is the number of wells in a plate. So anytime you see 96 things adjacently going wrong, that is not from the real world. That is from somebody mishandling, a plate, often just leaving it out on a bench top or something like that. So that brings up this question of like, how do we know that RNA is good? Again, not so much a huge concern for us now, but for most of the recorded history of RNA analyses, it was a pretty significant issue because the assays required intact RNA. Remember, the RNA is like a ribbon of copy of DNA. And the original RNA assays assumed that ribbon was intact because they were going to do things at either end of the ribbon, basically. Now we read off just a tiny little chunk of the RNA, just enough to determine where it came from in the human genome. So if there's a break someplace else down the RNA string, that doesn't actually matter so much. So the process of answering that question about, how good is my RNA sample often comes down to determining the size distribution of the RNA. So my messenger RNA that we're interested in is actually a sort of a widely variable size because genes themselves are widely variable in length. Some genes encode just a few amino acids, other genes encode, many thousands of amino acids. So the messenger RNA corresponding to these different proteins is a highly variable, essentially smear across a size spectrum. But we can arrange total RNA by size. So what you're seeing in the high quality RNA graph there is a size distribution such that small RNA molecules are on the left side of that graph and larger RNA molecules are on the right side. And you can see there's sort of 3 big spikes there, in addition to a sort of a grassy field of stuff between the leftmost spike and the middle spike. The grassy field between the leftmost spike and the middle spike, that's all messenger RNA that's going to encode proteins. The 2 big spikes are ribosomal RNA profiles. And ribosomes basically ribosomal RNA is not going to encode a protein, but it is huge. It's large amounts of it in cells, and it is physically very large. So ribosomal RNA species are a great way of determining whether RNA in general is banged up. It's hard to tell whether messenger RNA is banged up because they're not all the same size, but the ribosomal RNA species are all the same 2 sizes. So in the high quality RNA plot, you can see these 2 big spikes. That's what a healthy RNA distribution looks like. If you have some degradation, what you're going to get is an accumulation of debris and as kind of a shrinkage of those 2 big spikes to the left. So you can see how in the middle plot, the two big spikes have kind of moved to the left, there's like this, what we call scree. There's junk, basically fragments of RNA laying in the sort of the valley between the right two spikes. And there's this kind of shoulder of RNA trailing off. And you can see that even the messenger RNAs now are kind of like smaller as well. They've all moved to the left a little bit. In other words, there's a higher predominance of smaller RNA species that are consistent with damaged or degraded RNA. Highly degraded RNA ends up looking like what you see on the bottom, where everything has moved to the left. That is, all of the RNA molecules that used to be long are now, sort of broken up into little fragments. And so the mean fragment length is, a tiny fraction of the original length of these things. And you can barely see the two ribosomal RNA peaks a little bit there on the right-hand side, but most everything is in this kind of scree, sort of dust distribution on the left there. So once upon a time, degraded RNA like that, people just said, there's nothing we can do with this. This isn't going to work for our assays. Let's just throw it out. But, for you guys, just be aware that's correlated like 0.2, 0.3 with your socioeconomic status, often because of the conditions under which people were collecting the samples or things like that. This doesn't have to do with anybody's internal biology. It has to do with the pragmatic realities of, sampling in some of the environments that we care about. So, and by the way, that distribution on the lower right there is what salivary RNA looks like as well. So you can see why, the technologists don't love salivary RNA, because the salivary enzymes have, chopped it up into all of these pieces. We can actually assay that stuff reliably. We just can't interpret it very well. So I'll show you later why we can still get away with assaying now those degraded RNA samples pretty well until they become really mashed up. So you need extreme degradation at this point to have invalid data. But for most of the RNA history, if you couldn't pass the partially degraded, criterion there, then you basically just ejected the sample. All right, so that is RNA extraction and quality assurance. It's about 10:15 right now. Actually, I'll sprint for the end. I think we can probably do this here. So then you guys can have like a peaceful. Actually, do we need to do that? I'm going to not do that. What I'm going to do instead is not sprint for the end, because it turns out we have enough time to cover everything. And instead, what I'm going to do is open to any other questions right now so that we can have a soft landing before we go to the snacks, instead of like using up all the glucose left in our brains and forgetting everything once we eat a Twinkie or something. So yeah. Yeah, so there's some things that we use for validity checks that are, they're not particularly social unless you're into, the gender-related aspects of social. But X and Y linked gene expression we often use as, sort of just internal validation, like, do the self-identified males have more Y-linked gene expression? That should generally be the case. And in general, it's like huge, highly non-overlapping distributions. But every once in a while, you'll have somebody who, sort of is in the middle, or, somebody who's like flipped or something like that. So, nowadays that would be less puzzling to people, but once upon a time that led people to just decide, oh, let's throw this out because this is a, clearly poorly annotated or, something like that. So that's one thing that you could use as a kind of a ground truth experiment. In terms of the things that we tend to see, there are definitely some ancestry related differences in gene expression that reach this kind of ground truth level. So people of essentially kind of, Western European ancestry, broadly speaking, tend to have, let's see, I'm trying to remember, they have lower type 1 interferon gene expression than everybody else in the world. So, people of more recent African ancestry or people of Asian ancestry will tend to, all have higher levels of what we call type 1 interferon. So this is interferon alpha, interferon beta, again, salient recently because interferon type 1 interferon responses were the major gatekeeper of whether you get severe COVID in the first place. This like, well, totally, this is that they are the reason kids almost never show serious disease, because kids turn on really strong responses. Adults do progressively poorly over time. So that's another thing where, you know, demographically, you can see the correlation. What's interesting about the interferon system is it's very stress sensitive. So it's also one of the systems that is most often altered in people who are living in circumstances of adversity. So we will see, you know, easily like 40, 60, 80% reductions in type 1 interferon activity in white blood cells from people living in poverty, social isolation, that kind of thing. Really quite striking differences in many cases. So that's one. Another, you know, fairly reliable finding is more adversity tends to correlate with greater expression of inflammatory genes. So things like IL-1 beta, not so much IL-6, IL-1 beta, tumor necrosis factor, TNF, IL-8, COX-2, that kind of thing. So that's a fairly reliable, you know, finding. It doesn't happen literally every single time, but it's, you know, one of the most easily seen phenomena across lots of different studies. Yeah. Yeah, so the question is, what are the kinetics of you know, these effects? So that's actually a great question. One I should really have slides for to help people interpret. But basically, it takes about 30 seconds to maybe a minute to get maximal transcription factor binding on DNA after you've pinged a cell. So we know this because we can take white blood cells and drop norepinephrine on them, and then ask, you know, we can run the assays of these different biological processes at various time points. And what you can see is the signal coming in from the biochemistry is pretty fast, but nowhere near as fast as neurotransmission. So already you're like 10-fold off of what you would have in an MRI study. So it's very unlikely that you're ever going to come up with a tight correlation between the literally kind of, you know, milliseconds to seconds dynamics that you have in neural activity and what you're getting in RNA changes. RNA is incredibly sluggish temporally compared to those things. But it takes about 30 seconds for that initial transcription factor signal to start peaking out. That can last for a while because the transcription factors, I've idealized them as one transcription factor, but in fact, your cells are packed with transcription factor molecules, and they basically roll on, stick on the DNA, and then they fall off, and another one rolls on. So there's like this kind of stochastic occupancy dynamic that's taking place in these things. So that occupancy dynamic tends to wane within a few minutes of the initial signal if you don't keep pounding the signal. And a lot of receptor systems will stop listening to signal after, you know, a couple seconds anyway, through a process called desensitization. So that first wave is about, you know, 30 seconds to maybe 2, 3, at most 5 minutes for most transcription factors. Then you've got this RNA polymerase coming around. It's pretty fast, and there's a lot of those molecules. So it doesn't take long for those guys to get down onto the RNA, but it does take a while for them to do their work. So the transcription process takes 5 to tens of minutes to, you know, produce a full-length RNA molecule. And one full-length RNA molecule is not going to make a huge difference. So what you're really looking for is like, when does the distribution of newly produced RNA molecules start to really materially ramp up? When does it peak out and when does it go down? Usually, and this is variable across genes and across receptor systems, but the broad temporal kinetics are for a single pulse, the RNA abundance will ramp up about between like maybe, you know, it will typically ramp up over the course of maybe 5 minutes to 30, 60, for some things it's about maybe 90 minutes at most, but it will usually, in the absence of some kind of exception I'll talk about in a moment, it will start going back down, you know, starting at 30 minutes or 60 minutes or something. So the peak transcriptional response, we call it, depending on the gene, is like 30 to maybe 90, sometimes 120 minutes, but usually something in that range. And then to make protein, takes another tens of minutes. So it takes even longer for the proteins to come off of all of that. But at this point, these kinds of what we call first order effects get swamped by second order effects. So the second order effects have to do with feedback or stimulus persistence. So the dominant driver isn't actually how long it takes to turn these things on or how long it takes for them to spontaneously go off. It's how often you kick off positive feedback that keeps that system going without external stimulation. So positive feedback is ubiquitous in the immune system. It's not 100% positive. So it's not like when you turn it on, it'll go forever. But when you turn it on, it will auto-catalyze its own process for sometimes days or weeks. The other thing that happens that's important for social processes is that many of the social events that take place, particularly like psychologically, they happen very quickly, but they can become very persistent. So the dominant driver, for instance, if we do a monkey study and we reorganize the social lives of monkeys or something like that, you can see effects that last for 2 weeks to a month or something like that. But that's not because the RNA itself lasts that long. That's because the psychology, the social world around the monkey has led to a persistent change in their experience of life. And as soon as that change resolves, stops, or reverts to its original state, you can see again within a few hours to at most a day or two, they revert back mostly to where they were in the first place. So many of the kinetics at long time points are driven more by the kinetics of the psychology or the stimulus that's driving the RNA expression than the RNA itself. But the intrinsic kinetics of the RNA are actually pretty short. And that actually is a good thing, but it also creates volatility. It is, I think, okay, we got maybe 2 or 3 more minutes. One more question before our little break. Okay, 2 more questions. Yeah, that's true. So the question is, broadly, what about the disconnect between the duration of time for the phenotypes that we care about and the duration of time for RNA responses? So most of the phenotypes that we care about in health take decades to create, right? When you have a heart attack today, it's because you've been making an atherosclerotic plaque for the last 5 to 30 years or something like that. When you have a diagnosed cancer today, that cancer has typically been growing for a decade at least, maybe longer than that. And these processes, so you can think of as constructing disease at the tissue level. You're literally making a physical disease entity. You have to build the cells, or at least the structures that are created by these disease-producing cells. And yet these RNA processes are inherently pretty volatile. In other words, any pulse of RNA is going to produce, changes in transcription that maybe last an hour and a half. The proteins are going to be produced. How long the proteins hang around? I think the average, I think the half-life of proteins in the human body is about 80 days. So, every 80 days, you really have to replace a substantial amount of your protein. I mean, about half of it has kind of died off and needs to be put back. And so this, again, is the kind of the empirical representation of this fluidity I was talking about earlier, this constant self-regeneration that is going on and that this kind of RNA-mediated world controls. So this brings up this kind of disconnect between the relatively volatile kinetics of RNA and the relatively slow kinetics of disease development and disease recovery and that kind of stuff. And the key insight for how those two things actually fit together, there's 2 things that matter. One of them we call ratcheting. So ratcheting is when today I built 10% more of my atherosclerotic plaque, and now I'm going to rest for the next 5 years. And then, so there's this, but that atherosclerotic plaque is calcium. It needs to degrade very slowly to disappear. So if I make a lot of this relatively persistent calcium plaque today, it's going to hang around a long time. So it's, you can think of it as an extension of the 80-day half-life of the proteins. They have a long temporal tail once you've made them. Same thing is true, for instance, for neurobiology. We can grow neurons really fast, but the neurons don't go away right away when we stop pumping out nerve growth factor. So these things hang around and they can self-perpetuate. They can kind of auto-catalyze the production of the neurotrophic factors that keep them, alive. It keeps this neural fiber present in this environment. It will slowly withdraw over time, but much more slowly than the RNA goes away. So one of the disconnects is ratcheting this kind of temporal extension of a transient increase in protein production. The protein is much more durable than the RNA. The second thing is changes in what we'll call the basal regulatory regime. And this is, what we've been most surprised to be able to pick up in the world of social adversity is there seems to be a relatively persistent change in the baseline bias of at least certain RNA production systems. So as I was talking about earlier, one thing that seems to happen is that basally people just make, in general, less of these type 1 interferons when they're in threatened or adverse social conditions. They tend to make more pro-inflammatory messenger RNAs, even though each individual messenger RNA is degrading relatively quickly. The reproduction process has been bumped up to a sort of a higher pitch. So here, it's not the RNA that creates the temporal persistence, it's the change in the regulatory regime, essentially the transcription factor drive to this system, that is sustained. And it's usually sustained not at the level of durable chemicals, but at the level of durable cells. What happens is people adapt to circumstances, and those adaptations become cellular adaptations, like more nerve fibers in a particular environment, or more neurotransmitter receptors, or more neurotransmitter producing vesicles, or something like that. And it's that property that tends to hang on, and especially if it auto-catalyzes, it produces positive feedback. That's what seems to create these persistent temporal dynamics. So you can think of it as like juggling. The juggler can perform for a half hour, even though any ball is in flight for only a second or something like that. Does that make sense? Okay, one more question and then we're off to snacks. Yeah, so the question is, given that there's not, in any given tissue sample, there's not a perfect correlation between transcription factor activity, RNA abundance, and protein, how would you decide which one to look at in which particular context? So remember, it turns out that the protein is highly correlated with the RNA at the source of the protein. But in many cases, when we look at a tissue sample, it is not the case that the proteins that are there actually came from the RNA that is there. So this we see all the time in blood studies where there's like gobs and gobs of interleukin-6 circulating in most people's bodies, especially as we get older, especially if we're have higher levels of adiposity. This is like one of the most easily detected cytokines. And yet if you look at the messenger RNA level in the white blood cells that you pull out of blood, you'll find no messenger RNA for IL-6, 0. Like it's one of the most reliable things that we find. We, like in the beginning, we didn't believe it because, we just mistrusted the measurement technology. But, this is totally reliable. It's a real thing. And that's because the IL-6 that's circulating in blood wasn't made in blood. It was made in your spleen and your adipose tissue and other environments where there are gobs of IL-6 messenger RNA. So you can think of it as this, sort of circulating distribution from a factory that is not the factory that you're actually looking at in circulating blood. So that is the source of the disconnect. There really is a connect somewhere. The connect is actually pretty tight. It's just that it's not guaranteed that the factory is in the same place as the cytokine. So Then to answer your question about, well, then which things should we look at? The answer depends a lot on the context that you're trying to understand. Like, let's say I'm trying to understand an atherosclerotic plaque and why it's big. One of the things we know about that is that monocytes are the major cellular, sort of, if you will, arrivals that then construct more atherosclerotic plaque. They transform into macrophages and they do all of this wound healing stuff that as a side effect produces this calcium scab that was supposed to be healing this wound on the side of your coronary artery, but it's now getting bigger and bigger and bigger and blocking off oxygen to the muscle. So I could go to the coronary artery and literally take samples of that. But nobody likes that because then people are going to have heart attacks. And also if the plaque was there, somebody should have taken it out before the researchers got to it, right? So there's various reasons to suspect that won't happen very often. But I could say, those monocytes, like if this person's building a plaque faster than that person, maybe that's because the monocytes that are coming in and then converting into macrophages to build this thing are different somehow. So I could look at the blood monocyte pool, knowing that that is going to be a significant chunk of the arrivals that will get into the sort of the atherosclerotic plaque itself. So a lot of it has to do with learning what we, I guess would call pathogenesis. In the context of disease, we would need to come up with an ontogenesis in the context of development and behavioral genesis and stuff like that. But you need to have some kind of a theory about who is actually making the phenotype that you care about, the outcome that you care about, and then trying to see if you can sample them, even if you can't sample, let's say, the brain tissue that's the proximal determinant of behavior. All right, that's it for now. Have a happy break. We'll see you guys, I think, in about, 15 minutes? Is that what it says on the agenda? It's 10:36 now, so I'm going to give you guys until 10:50. Does that sound right? Yeah. Those of you who came in late, I'm going to let you know the Wi-Fi settings for the meeting attendees. So for those of you who are like borrowing somebody's room Wi-Fi, it's, hey, Michael, I'm not sure that the PA is working at this point. Is that right? Did I get disconnected or do I need to use this one or no? Sorry, you guys. Okay, there it goes. So for those of you that came in late, this is the internet connection that they'd like us to use, password LCC 2022. If you have any additional questions, let us know. Sorry for the animations here. It's going to be tedious, but almost done. So what we're going to pick up now is essentially, now that we've got RNA from someplace that makes sense for our purposes, how are we going to measure that RNA, and particularly, sort of in a format that allows us to do that at scale? In other words, for all 20,000 or so human genes and the various alternative versions of each of those genes transcripts. Transcripts are made in little chunks, and you can actually swap out chunks or sort of skip chunks here and there or add in other chunks here and there. So that's what allows a given stretch of DNA to encode multiple what we call isoforms of RNA. But that triviality aside, the bigger question is, like, how do we measure the abundance of particularly the messenger RNAs that we're interested in a particular tube that has, water with RNA floating around in it? So these days, the dominant way we do that is through RNA sequencing. There's another assay that was pretty ubiquitous for the first maybe decade or so of the century, microarray assays. In some respects, actually, I miss the microarray days because they had certain properties that actually made it much easier to apply standard measurement theory to them than RNA data. We'll talk about the quirks of RNA data a little bit before lunch and even more so after lunch. But unfortunately, the world of microarray-based RNA profiling is receding because it is so much more profitable for the people that make both of them to sell RNA sequencing equipment than to sell microarrays. So I'm going to skip microarrays for now, but if any of you are interested in it, it basically at the question and answer session before lunch, just raise your hand and say, hey, I actually have microarray data. Can you show me, what that means in terms of measurement, how that's done, and numerically sort of how it produces different kinds of data than what RNA sequencing would. I'd be happy to do that. So the core idea is that we have RNA that we're interested in floating around in the water inside our tube. And we are going to use an enzyme that converts that RNA, basically an enzyme that reverses the normal transcriptional flow of information from DNA to RNA. We're going to take some tricky enzymes that actually convert RNA sequences into DNA sequences. And we are going to run that reverse transcriptase, they call it, down the RNA molecule. And every place on the RNA molecule where there's a T right now, it's going to deposit an A. Every place there's a C, it's going to put a G. Every time there's an A, it's going to produce a T, so on and so forth. So it's just going to create a base pair complement copy of the RNA that is in the, floating around in my sample fluid. Now, in the original version of RNA sequencing, they did that for all of the RNA that was present. But remember, there was those big spikes of ribosomal RNA. The vast majority of the RNA floating around in the fluid is actually not anything that's going to encode a messenger RNA that then becomes a protein. It's going to create, these big protein factories called ribosomes. So, there's various versions of this. For a while, they were like trying to kill all the ribosomal RNA, but you have to be incredibly efficient to do that, or there's so much ribosomal RNA, it will still swamp the messenger RNA. But later, they came up with a chemistry trick that, again, in biology, we've been doing for years, which is like, actually, you can distinguish ribosomal RNA from messenger RNA that encodes, a protein by virtue of these little tails of A's at the end, what's called a polyadenylation site. So a polyadenylation site, A strings at the end are pretty ubiquitous. Almost all messenger RNAs have polyadenylation sites. So you can actually use enzyme systems that only reverse transcribe polyadenylated RNA. So only the messenger RNA is going to get converted. So you're not going to convert all of this stuff that you're then just going to try and throw in the trash later. Or will overwhelm your assay platform. So we'll get messenger RNA specific reverse transcription to produce this so-called complementary DNA sequence. So you have a DNA sequence created by the reverse transcriptase enzyme that's a complement to the original RNA. And then, as you guys all know, we have great DNA sequencing infrastructure from the Human Genome Project, basically, that we can just feed this DNA to produce measures of the abundance of specific RNA molecules that produce those cDNA representations. So without going into too much detail, and I'll show you guys a link to some videos later so you can see how this kind of stuff is done in actual practice. But there's a number of different chemistry strategies for taking these big long strings of complementary DNA and interrogating them. Usually those interrogation processes involve copying, again, a fixed amount of sequence. So, maybe you will use a system that copies 100 nucleotides of information, which is about at least 50 nucleotides more than you really need, but whatever, it's a lot better than running thousands of nucleotides. You're going to copy 100 nucleotides of information from that complementary DNA. Originally, they did this by randomly landing, these copying machines all over the DNA strand. But later, they decided if we're really only interested in the messenger RNA, let's always land these things at the polyadenylation site and then read in about 50 or 100 nucleotides worth of information in the essentially the coding non-polyadenylation region of the messenger RNA molecule. So we're going to actually focus in using this highly selective base pair complementation chemistry. We're going to focus in just on the messenger RNAs that will then structure proteins. And for each RNA molecule, we're only going to read the 100 or so nucleotides closest to the polyadenylation site. What that means is I'm not going to read nucleotides from the rest of the many thousands of nucleotides that might be present in that complementary DNA. And that turns out to make things vastly more efficient later, because I'm not going to be collecting reads, 2, 3, or 10 reads from the same RNA molecule. Every single RNA molecule that's in there is going to generate only one read, which means I can actually pretty accurately count the number of RNA molecules for any particular gene in ways I'll show you in a minute. By virtue of the fact that I know each molecule I'm making only one copy of, or one sequencing read from. So in any event, we'll use some strategy to make these limited length sequencing reads, and then these things basically end up going into files of literally ACTG, and then often there's like some quality of read assessments appended to those things, but these are called FASTQ files because of the format. The FASTQ format basically, if you're interested in it, there's a file on the Google Drive that actually walks you through the sequence of files produced in RNA sequencing. So the very first file is a FASTQ file just showing you the first whatever 3 or 5 reads from some particular sequencing assay, and then every step downstream that we talk about, there'll be another file that gets produced by virtue of, let's say, feeding that FASTQ file into this sort of data production process. But the very first thing we need to do is, we've got these reads that start with this polyadenylation site and then are like maybe 100 nucleotides worth of information. 100 nucleotides, even 50 nucleotides, probably about 12 is enough to uniquely identify the location of any particular gene transcript on the human transcriptome. Usually we look it up in the human transcriptome because it's a great deal more DNA in the whole human genome than actually gets transcribed. So usually we're not interested in the intergenic regions, the regions between coding regions. And so we do this lookup process only on the collection of messenger RNAs that actually, only on the collection of DNA sequences that actually get transcribed at some point or another. But in any event, we've got the selection of potentially transcribable DNA in a dictionary, a database from the human genome. So this is like, genome sequences from either the original sequencing effort, or now, there's many thousands of alternative, sort of human genome sequences that you can use as well. But typically we use what's called the consensus or the reference human genome sequence that is, sort of essentially adjusted out to constitute what they believe to be the most prevalent version of the human genome. It turns out the minor variations in genetic sequence that are ubiquitous and allow us to do population genetics don't surprisingly make very much of a difference for this transcript localization and counting process, because it's highly robust to 1 or 2 or even a few nucleotide aberrations or mismatches. So that's one of the advantages of having more than those maybe 12 essential nucleotides, is if a couple of those nucleotides are messed up either technically or due to real genetic polymorphisms that change the coding sequence of the messenger RNA. This so-called mapping process of counting transcripts from particular regions of the genome will tolerate a certain amount of local mismatch. So that basically increases the likelihood that each of these reads will be located somewhere on the human genome. And usually about 90% of the reads can be found on the human genome. The remaining 10%, people once upon a time were very worried. They're like, 10% of our stuff is mystery. It turns out 10% of this stuff is messenger RNA from microbes or other kinds of weird things, and probably a much smaller amount is truly goofed up, technically invalid sequence. But in any event, each of these reads, we are now going to skate through the transcribed subset of the human genome and ask, does this read match most closely gene 1, gene 2, gene 3, so on and so forth for 20,000 genes, ultimately probably 50,000 or so distinct transcripts. So in this particular cartoon example, we have one read and we found, oh, this read matches the OAS gene better than it does any other, sort of transcribed region of the human genome. So that's one read that we say is mapping to the OAS gene. In other words, it matches pretty closely, much more closely than you would expect by chance, and much more closely than any other region of the human transcriptome. So if we do this over and over, we typically acquire at least 5 million, typically about 10 million reads from any given RNA sample. So there's about 20,000 genes in the human genome. So you can kind of do the math. It's not a uniform distribution. Some genes are expressed at very high abundance. Other genes are, as we'll see, are expressed at zero abundance. And probably only about half of the transcriptome is actively expressed in any given cell at any given time. So that means that with between 5 and 10 million reads, we'll typically be able to come up with a reasonably accurate quantification of any gene transcript that has an abundance of more than, let's say, 1 in 10 million, between 1 in 10 million and 1 in a million. We'll be at limiting dilution around 1 in 10 million, and we'll be at about 10 counts for things that are present at 1 in a million prevalence relative to all the other transcripts in the cell. So that doesn't get everything, but that gets most of the things that are, reasonably frequently transcribed. And you can always, if you're interested in getting more precision, you can acquire more reads, but there's like, it's almost like a power curve. Like at this point, it becomes, linearly more expensive to acquire like 20 million reads than 10 million reads. But the number of genes that you can more accurately, quantify at this point is probably only like 2 or 5% at the most, or something like that. So usually the sweet spot is somewhere between 5 million and 10 million for big population health studies. If you're an RNA biologist and you're really interested in these transcripts, they'll do like 100 million reads from 2 experimental samples or something, because they really want to get this stuff exactly right. But for the kind of stuff that we do, where we're mostly just interested in sort of the conceptual equivalent of an ELISA or something, measuring the amount of IL-6, turns out about 5 to 10 million reads total is sufficient. So each one of these reads, we have now found a spot in the human transcriptome where we think it came from. So in this particular case, I'm using the IL-6 example. There's 1 read that mapped to IL-6, 4 reads that map to OAS. Every single gene in the genome has some count of the number of sequencing reads that ended up matching that position of the transcribed human genome better than any other transcript. And there's, again, a batch of maybe 5 to 10% or something that just, we couldn't find a match anywhere, either because they came from microbial RNA, or maybe they came from, damaged, like broken, some weird thing that afflicted the chemistry up at the top. So that's kind of typically what you'll see. One of the things to note now is that the reason that this kind of polyadenylation targeted messenger RNA sequencing is so robust to RNA degradation is that this actual RNA transcript at the very top goes out to the other side of the football field there. Like it's thousands, sometimes tens of thousands of nucleotides long. What RNA degradation is basically is a physical break in that string. So if you're going to rain a random physical break in that string kind of uniformly over the distance of this transcript, the odds that you're actually going to mess up the first 100 nucleotides are really small. So that part, even when you get massive degradation and lots of breaks, usually you haven't broken it so severely that you don't have a stretch of 100 or so nucleotides attached to the polyadenylation tail for that assay to pick up. So that's why you can get totally banged up RNA. And actually, these days, if you're using these kinds of assays, you can still read this stuff out with pretty high accuracy. This is also what's made it feasible to work with dried blood spots, which the drying process does break RNA, but it probably breaks every strand, maybe 2 times or 10 times or 20 times, but not enough to actually break it so much that it damages the so-called 3' end of the messenger RNA molecule right adjacent to that polyadenylation site. So that's what we're doing. So you guys will hear about reads and read depth. Read depth is just the total number of reads acquired per sample. You can, sort of increase read depth if you want more precision at weakly expressed transcripts. You can reduce it if you don't have a lot of money. That's actually a great strategy to kind of walk it up and down. Often for the kinds of things we're doing, it's more important to be able to do everybody in your data set, all 20,000 people, than to come up with incredibly precisely accurate measures of all 20,000 genes. So you might totally reasonably decide, I want to do more samples, but less sequencing depth, because, you know, I already know based upon pre-existing data or something like that, that I'll have enough sequencing precision to, you know, sort of catch most of the transcripts that are present. I know I'll miss, you know, a small number of weakly expressed or highly variably expressed transcripts, but I'm a population scientist and I really care about getting all 20,000. I don't want to do 10,000 at the depth that my sequencing core is telling me I should, because they get rich from that. I want to do 20,000 at half that depth, basically. So that's the kind of stuff where, you know, as you go to acquire data, it's definitely useful to talk with people who share your substantive interests. Don't necessarily just go to your local genome core and say, what should I do? Because they won't do 3' sequencing because they don't make as much money off of that. And so you'll be able to assay 5 samples instead of 50. Like often that is the magnitude of the difference. If you just go into a sequencing core and they say, hey, this is our gold-plated Illumina standard here or something like that. So be aware that there's lots and lots of optimization that you can undertake at this stage that will allow you to make population health research happen on population health research budgets, basically, as opposed to the crazy budgets from the Human Genome Consortium or something like that. Yeah, so the big population health studies definitely are going to have variability in what we call apparent depth, which is the total number of reads acquired. They're surprisingly close to one another, in part due to financial constraints, because the ones that tend to acquire more reads also acquire those reads from what we call full-length sequencing, as opposed to this kind of restricted messenger RNA specific sequencing. So the actual total number of effective counts, they're probably all headed towards about 10 million or so, something like that. And that's because if you do the kind of the industry standard Illumina sequencing, it's only, you know, you're basically losing about 4 out of every 5 reads to, you know, sort of multiple reads on the same transcript or stuff like that. So they're broadly speaking closer to one another than you would expect, but they're not going to be perfectly validly poolable because of differences in the actual read generation process. So in general, most of us, you know, in the industry would recommend meta-analytic integration across the studies, not trying to kind of physically pool counts across studies. You know, estimate your models within each study and pool the regression coefficient somehow, as opposed to trying to treat them the same, because they do have different stochastic properties. And the deeper the depth, you know, the more a small number of transcripts will be resolvable. Ironically enough, on our call with Bharat, he just says, I'm going to squash all of the low-level transcripts for our, you know, whatever, our default working data set. But in the raw count matrix in HRS, you know, you'll be able to dive down pretty deeply if you don't kind of squash up and floor up to, you know, 10 reads per million or something like that. Okay, so this, you know, everyone's always saying, can I come see your lab? Can I come see the sequencing lab? And I'm like, all labs look the same. They're all just gray plastic boxes. You know, it's like, so that's, you know, the industry standard sequencer right now. It looks suspiciously like the RNA extraction robot I showed you earlier. It's like really quite anticlimactic to see these things, but that's sort of a good example of what these machines actually look like. What you basically see is like that little, you know, sort of rectangular kind of border there in the very middle is just like a little hatch that you drop a plate into, and then amazing magical things happen. But you know, you never really see this stuff because human beings are not coordinated enough to do this thing. But essentially what this particular version of sequencing does is it has a fluorescent molecule that it has basically fluorescent A's and then G's that are tagged with the different fluorescent molecules, and then C's that are tagged with yet a different fluorescent molecules. And I can't remember which ones I did. A, C, T, and G all are tagged to different fluorochromes. And they're going to land each one of these complementary DNA strands at some physical place. And they're going to run a bunch of cycles where in each cycle they add one nucleotide to the copy that's being made. So each nucleotide that gets added produces a little fluorescent signal. So there's a very sensitive camera. It's basically, sort of at each pass of adding another set of nucleotides, it's going to take a picture to see which color nucleotide got added at this physical spot. So this physical spot, it's basically sequences of keys are what landed. You can think of conceptually on the sequencing platform. When it's physically there, it's going to be like red, blue, green, red, blue, green, 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 red, blue, or something like that at each cycle. So that's literally the information that gets read out. So that thing is mostly a camera, it turns out, more than anything else. It's a very, very good camera, right? It can pick up basically, I can't remember the number of photons, but it does not a lot. It's incredibly sensitive, and they've got a huge number of molecules parked in these so-called flow cells that are the things that, are the platforms that you're building these fluorescent copies on. So anyway, this is the dominant, they call it sequencing by synthesis strategy run. You hear the company Illumina really kind of popularized and sort of effectively drove the market to prefer this sequencing by synthesis. And it works pretty well. It's pretty cost effective. You can generate much better chemistry to produce your input complementary DNA than Illumina will sell you, because Illumina doesn't want to cheat themselves out of the cost of the revenue from their perspective of the supply. But basically, all of that, once you've got these complementary connected, once you've got those complementary DNA sequences connected to the platform, it doesn't really matter where they came from. There's adapters that will allow any complementary DNA system to land onto the Illumina sequencing platform. So that's what the sequencers look like. There is a whole flow chart about how to choose the complementary DNA synthesis strategy. Once upon a time, I walked people through it, but it's kind of tedious, but it's not a bad idea if you're starting off a study and you don't just have mountains and mountains of money, so money is no object. If you're heading toward the assay phase of the study, it's not a bad idea to check in with somebody who knows about the interests of population health researchers and does this kind of thing to see what the latest options are, because these things change continuously over time. They get much better. And if you go with the default option that made sense 5 years ago, it's going to be more expensive and you're actually going to get less data, oddly enough, for spending more money on it. So I won't, sort of bore you guys too much more with this, but between the cost profile and its ability to deal with partially degraded RNA, there's quite a lot of strategy that goes in this space. So not a bad idea to kind of optimize that real time as you're getting ready to go to assay. Okay, let's talk about what you get out of this whole thing in terms of a data file. But actually, before we do that, maybe I'll take a little break here and see if anybody has questions about that kind of basic chemistry and complementary DNA strategies and sequencing depth and stuff like that.
If you want to look at protein differences or
so the question is, if you're interested in alternative splicing or microRNAs or other things that don't have a polyadenylation tail, then you wouldn't use this approach. That's correct. In other words, this approach will only allow transcript abundance counting. So the vast majority of population health researchers don't even know what to do with alternative splicing data. But if you are the small minority that's like, I want to know, let's say, about the glucocorticoid receptor beta isoform relative to the alpha isoform or something like that, then you have two approaches. Either you could try sequencing everything and collecting the universe of those things, or more efficiently, you could just go do a PCR for that kind of stuff on it. It will turn out to be much less expensive and much faster to do an isoform specific PCR specifically on what you're interested in. So the only people who would really benefit from transcriptome-wide sequencing data would be people who are interested in discovering new splices, basically, in transcripts they had no idea to look for in the first place. So it's not impossible, but it probably won't be it will be only a tiny minority of the likely investigators, and will drive cost sort of geometrically greater to pick up that kind of information. Any other questions?
Hints of a process, I mean, not at the measurement, but right at the recovery.
Actually, even the measurement as well. Yeah. So keep going.
Is there
Yeah. So the question she's asking is, how are you going to deal with questions about quantifying validity here? Should you run duplicates? Are there other ways to do this kind of thing? So there's 2 or 3 different versions of this. One of them is I'm going to run literal duplicates. Another version, and typically they don't do that for every sample, but they do that for some healthy subset to try and quantify the magnitude of the noise. But there's another crowd that says, I'm going to run duplicates of every sample just by increasing my sequencing depth, right? So they're going to say, oh, I want, the question you're really asking is about the quantifying the precision of, sort of the measurement in a kind of a measurement reliability sense. So some people say, oh, actually, I'm just going to try and drive that causally to a better point by investing more money in sequencing. Other people will say, I'm going to do that by getting rid of sequence reads that don't inform me about the thing I'm interested in. So those are the people that would, let's say, do the 3' stuff. None of these people will do no duplicates. Everybody will do duplicates at some point to just try and quantify where they're at. But the downstream quantification actually becomes a non-trivial philosophical problem. Like, say, if we're looking at one readout like IL-6, it's very easy. Like, okay, I'm going to compute, whatever, a test-retest correlation or a coefficient of variation or something like that. But I've got 20,000 genes and 50,000 transcripts. Am I going to compute the average? Well, actually, the average, or median or whatever are, sort of, even then, that's like, it's not going to be a very good representation of the whole distribution. So early in this kind of process, they then started looking at transcriptome-wide representations of the distribution of these things, and asking about they would come up with essentially CV distributions and try and optimize those kinds of things. So you can do that. In the end, most people decided that they couldn't really do anything other than describe, this is where we're at for better or worse. And that most people took their attentions and instead just devoted them towards trying to do better at what they can. So most people don't run duplicates on everything, but most people do run duplicates on a small number of things. There's definitely cultural variation in this, like immunologists by program, like in their DNA, they're like run triplicates of everything, basically, is their mindset. Whereas, in behavioral sciences, we might be much more inclined to do, duplicates or triplicates on a small number of things to estimate where we're at, and then to use that information quantitatively in the data analysis. Like, let's say we plug that into a structural equation model as a reliability estimate or something like that. But the challenge is that you're going to have a CV or a reliability coefficient for every one of those 20,000 genes, half of which are not expressed at all, right? So your reliability coefficients for them aren't going to mean anything. There's going to be another half, so another quarter of the total, where they're expressed at very, very low levels. And it's unclear how much true signal there is. And most reliability coefficients are a ratio of true variance to measurement variance. And so if the true variance is very small, they're always going to have crappy reliability, not because the measurement technology per se is so bad, but just because there's really no signal there, right? It's very hard to come up with a correlation. So it's really only going to be about maybe 10 to 20% of the total transcripts where we feel, and you can experimentally create variance, but in population health studies, we've just observed variance that's kind of ecologically there. So it's really only going to be about 10 to 20% of the transcripts where you can come up with what I would call a sort of the idea of a substantively meaningful reliability coefficient doesn't sound right since it's a technical parameter, but basically it's going to be only a small subset where you can really go like, okay, I can trust this reliability coefficient to tell me something about this. There's true score variance here that is, real, and therefore I can ask something useful about the ratio of noise to true score variance. Yeah.
I'm assuming we actually have done a really good job putting the samples without like.
Right. Right.
That was done.
Right. So the question is, in the spectrum of technical vulnerability or technical noise, where does RNA assay fit relative to, DNA sequencing where it's relatively low concern versus methylation assays? So methylation assays, are done predominantly by microarrays still today, and microarrays do have big batch effects. And so, they're going to be an extreme version. RNA had similar batch effects when it was being run predominantly by microarrays. Sequencing has much smaller batch effects if done well. So, your question was like, let's just say we collected the data as well as we could. We were careful not to degrade whole physical plates of stuff like that. And so let's just talk about intrinsic assay performance. So it turns out even the assay, like, the robot is not going to do all, we do, 20,000 sample studies, right? So the robot, that's a lot of work, even if a robot's doing it. So in the end, the way people deal with this is they certainly do post hoc batch analyses. And I can tell you from the RNA sequencing, batch effects are much smaller in general in good hands. The batch effects are huge though, if you don't think about some fairly simple things that are super easy to avoid. Like, don't send half of your RNA to Berkeley to get sequenced and the other half of it to UCLA, right? That's just a no-brainer. But believe it or not, people do this all the time with something. And of course, they send everything from this condition to Berkeley and everything from that condition to UCLA. And it's like, what are you doing, right? So as long as you do reasonable methodological practices, batch effects are very, very small in this. The other thing, though, that even swamps the batch effects is the cDNA prep. So you're going to get structurally different results for 3' sequencing of just messenger RNA versus transcriptome-wide, everything versus transcriptome-wide trying to eliminate ribosomal RNA. So those things, which are not technically batch, but end up being picked up by many batch effect analyses because people just aren't kind of thinking in this more refined way that we were describing. So the good news is that sequencing is incredibly accurate, but the bad news is it's so accurate, it reveals a lot of stuff in the stochastic world that we just couldn't see before. So microarrays will always produce some fluorescent signal, right? It's never going to be zero. It might be truly zero, but it's going to have a, sort of a stochastic spray of fluorescence noise, essentially, camera noise, you can call it on that. These things, they're crazy accurate. And so you're going to get counts that are literally 0 or 1 or 2. And if you go back and reinterrogate this thing, you're going to get pretty close, it turns out, in a lot of cases. So, when you're working in the count world, something that in one run gets you a count of 1 and the next run might get you a count of 3, but it's usually not going to get 1 and 300,000 or something like that if you're actually doing literal physical replicates. But if you take blood from one arm at the same time as you take blood from the other arm, you're going to get huge differences. And that turns out to be about biology, not about the technology. So the good news is intrinsically, the technology is pretty tight as long as you do good methodological practices. But in practice, it's still hard to avoid, intrinsic biological noise. So the reason now to do batch effect analyses actually isn't, I mean, part of it is, like we still do batch effect analyses on all of these things. We still find plate differences, but we don't think those differences are coming predominantly from the chemistry under ideal circumstances. We think they're coming from what we call hands in the biology world. Like there's just big differences in how scrupulous different people and laboratories are. And so we don't want to name names about, who, but we have definite places we like to work with and definite places where it's like, you can do that if you want. So that's the big driver right now of batch effects is hands and the good sense stuff you were talking about. Like don't send all of one condition to UCLA and all of the other condition to Berkeley. You confounded, condition with the assay. Okay, any other measurement technology questions at this point? Great. All right. So this is what we're going to get out the back end of this whole thing. This is the transcript abundance matrix. Transcript abundance matrix conventionally are structured with genes in rows. So I've got gene 1 through gene 20 if I'm quantifying transcripts at the level of gene, or maybe I've got transcript 1 through transcript 50,000 if I'm quantifying at the level of the alternative isoforms of the transcripts. But for each sample in my data set, I've got some count of reads that align to this particular segment of the human transcriptome. So that's this is a cartoon model, but it illustrates some of the key things that you'll typically see in RNA sequencing data. First, there's a lot of inter-subject heterogeneity in some genes. In most genes, there's not, but in some, it's like really crazy. Like gene 1, you're like clunking along at 2 digits, and sometimes, and then you're to like 25,000, and then you go back to 2 digits or something like that. That's real, it turns out. Like there is something going on in sample 4 that just, is atypical relative to these other people. And that turns out to be real biology. But that is not, linear models do not love that, right? So this is going to be something that we need to contend with in a way pretty much newly. And it is as when we're in the microarray world, this was, you could still see it, but it was much blunted by the much poorer signal to noise ratios. But because sequencing is so precise, you see these crazy things that are coming from real biology much more often. The second row gets you to about 50% of your experience in the transcriptome, which is like, this gene is there, but I have no reads for this in any of my samples. It's all zeros. So that you have to kind of think about philosophically what you want to do in the long run. For most of us, it doesn't matter because you can look at those zeros and say, this is not something that correlates with my outcome of interest. But there are times when people are like, I want to know about IL-6, it's all zero. But that like there's all this IL-6 protein, like it's all zero. The cells that are circulating don't make that IL-6, but still, just be aware that the zeros, will require some defense or explanation sometimes. Row 3 is another example of what we see a lot, where you get a lot of zeros, but a few people have non-zero reads. In most cases, we speculate, rarely do we actually directly verify this, but in most cases, we think this is what we would call limiting sequencing depth, and that if we doubled or quadrupled or tenfolded the number of reads that we collected, those zeros would actually be, sometimes 1 or 2 or 10 or something like that, and that we just didn't sequence deeply enough to pick up this particular gene. So we call that a weakly expressed gene or a variably expressed gene. We can't tell yet. We can't tell whether those are true zeros in some people and true non-zeros in others, or whether they would be all non-zero if we just sequenced more deeply. But on the other hand, we're not made of money either. And so at some point, you just have to stop sequencing. So you'll always have some genes that are at that limit. And you'll need a data analysis approach that can deal with a lot of zeros and then a bunch of non-zeros in some kind of systematic way. And then gene 6 is another example. There's huge differences in the general abundance of different transcripts. So the vast majority of transcripts in the human genome are expressed at relatively low level. Again, like I said, in any given sample, about half of them are all zero. Another quarter of them are many zeros, maybe mostly zeros, but a few scattered examples here and there, which might be suboptimal sequencing depth, or it might be true individual heterogeneity. And then there's a very small number of transcripts that are expressed at very, very high levels in this kind of exponential curve. And so that structure means actually that the vast majority of your sequencing reads are being consumed by about 2% of your genes at the most. And so trying to deal with that is an issue because often those highly expressed transcripts are not of great substantive interest. They're transcripts that are involved in creating cell structure. Sometimes if you're a structural person, maybe that's of great substantive interest, but most of us are like, we don't think that those highly expressed transcripts in general contribute to most of the behavioral or disease phenotypes that we're interested in. So it's a little bit of a pain to lose, so much of our total sequencing resources to these highly expressed transcripts of, sort of questionable substantive significance, but that is the reality of the biology. And we can't really go suppressing those things because it will limit our ability to do certain kinds of normalizations later. We need to count everything that's there in order to optimally normalize the more weakly expressed things, which will often actually reduce some of these extreme count disparities across samples within some of these genes. So anyway, that is what transcript abundance matrix will look like. So it's got some features. One, the number of genes is typically much greater than the number of samples. The only place where in population health, I think our number of samples is approaching the number of genes is with HRS, but in most other places, you're never going to throw that whole matrix in and, it's not going to get inverted in some analysis, right? You're going to have to do some processing to reduce feature space, as they call it, reduce the number of variables being analyzed. You can't just swallow that whole thing in one fell swoop and then kind of extract magically the answer from that the way you could like inverting, an XY matrix in regression or something like that. So that's one. The second is these are count values. So they're going to have a stochastic structure, noticeably different than what we're used to, continuous variable, sort of normal distribution, general linear model style. So that has led a lot of people to embrace literal count-based data analysis, which turns out to be a terrible, terrible idea because although they are in their native form counts, the moment you normalize them and the moment you try and take out any kind of technical variability, they lose their official count-like structure. That doesn't mean that they lose all of the stochastic characteristics that come from count stuff, but no longer are they officially counts. But the real reason not to run count-based analyses is that count-based analyses usually make very strong assumptions about the relationship between the mean and the variance, either that they are the same, let's say, in some models, or that the variance is proportional to the mean. That's the part where, in fact, count-based models are like easily shown to be inaccurate for this stuff because you can easily find genes that have the same average count level, but vastly different levels of intra-individual variability. So it's really not the count structure per se, but people got very excited about counts. I get to go back and show everybody I remember my statistics, it's like okay. And then, most count-based analyses, in fairness, count-based analyses can be made every bit as flexible as, continuous score analyses using the, sort of the magical world of statistics that we have now. So it's very easy with generalized linear statistical modeling to contend with that kind of stochastic structure that comes from counts using very sophisticated designs with, covariates and cross effects, everything that you're familiar with from linear modeling. But it turns out almost nobody goes to check and see whether the residuals actually match these assumptions that the count-based models have. I've never seen them pass. So I've never seen like a straight monotonic relationship between mean and variance. I've never seen, plus or minus like one or two, sort of counts on these kinds of things. It behaves much more like normal stochastic data. So it looks like counts coming in, but it behaves in terms of the residuals that actually determine, the validity of your p-values, essentially your standard errors. It behaves much less like counts than people would think. You can treat it like interval data, yep. Or you can treat it as, I mean, most people just kind of throw it in as, yeah, random, like they fit a normal regression model or a t-test or, all of that kind of stuff. So yes, it's more than okay. In most cases, it's demonstrably preferable to treating it as count data because the counts bring along these very strong assumptions. And these assumptions are so extreme that they produce insane p-values as a result if they don't match the. You could treat, yeah. So are you talking about treating the numbers as nominal? You could, but you would use up a lot of degrees of freedom. So like I have a 25,609 or something. Like I don't think you want, 30,000 nominal categories. So you could do that under some restricted circumstances, but in most cases it just wouldn't be practical. Yeah. Okay, so one of the issues that we need to contend with is there are many zero values, including genes that have all zeros. Most of that stuff will actually take care of itself, but if you just plug it into stuff without thinking, if you take all zeros that have like a 1 here and a 4 there, you can imagine, crazy stuff happening, right? So just plugging these vectors of expression data into analyses iteratively over genes without thinking about this kind of stuff. I mean, sometimes, the system will say, I can't deal with what you just gave me. It's, all zeros, so there's no variance or whatever. That'll be easy. The part that you miss is when it's all zeros, but maybe a few, small non-zero values here and a few small non-zero values there, and then 125,000 there. And you can, get fabulous p-values from that, but it's very questionable whether that will replicate or whether, it's very easy for systems with data like this to essentially to overfit or chase outliers or that kind of stuff. So one of the other issues is the sparsity, the non-zero values being here and there, but not everywhere. But heteroscedasticity, also huge issue. As you can see, some genes, very high expression levels, they're going to have very high variance. Others, just barely, they're going to be all zero, which is actually going to look like you have incredibly consistent data, right? I mean, no measurement error, it's 0, 0, 0, 0, 0, 0, right? But that's actually not really the case. There's no variance because you can't see the true variance most likely. So paying attention to those kinds of issues is going to be important no matter what you do. So we'll go through all of those things in the afternoon and in some, sort of general detail and talk about the consequences of that. One thing that is worth doing early on, though, is finding cases of data or columns of that transcript abundance matrix where we just don't trust the numbers. So I didn't really show an example of this kind of thing here, but one of the most common is, you'll be looking through a transcript abundance matrix like this, and then you'll see, like, especially if you sort the data so that the most highly expressed transcripts are at the top of this. So, gene 33257 or something like that, it's at the very top because it's the most highly expressed transcript. And you'll look across and you'll see, this thing is in like log2 transformed units. It's like, 14 or 15 or 20 or something like that. And then you'll see something that's like 0. And you're like, no, that's not right. There's just no way this thing can be 0. And so you'll find a lot of samples where there's a lot of zeros vertically in this matrix that just don't look like the pattern of zeros that you see everywhere else. Seeing zeros for whatever, gene 10, not a problem. Seeing zeros for gene 6 will make you scratch your head, right? You're like, I don't think so. Even seeing 100 for gene 6 would make you think that there's something wrong. It turns out there's usually a pretty strong correlation between aberrant data on one gene and aberrant data on other genes. So we'll normally want to try and identify those samples and then either throw them out just because they look completely insane, or at least think very hard about this sample and whether it's something you want to analyze. Sometimes that's going to happen because that person has leukemia and they have crazy blood and it looks completely different than everybody else's. And so you just have to be able to answer for yourself the question, is it useful to have a leukemia patient in my sample, right? So if it is, if you're like a biomedical researcher and this is part of the spectrum of the biology that you're studying, maybe it's okay. If you're mostly interested in how social factors, influence health risks for people in general, not for leukemia in specific, and this is going to produce this crazy outlier that's going to start dragging around all of your statistical estimations, then you might decide to get that thing out before it causes a lot of trouble. So it's up to your substantive purposes when to prune those things out. Sometimes it's very obvious that this stuff is just technically invalid, but other cases, there's weaker disparities that are true biology, but maybe not the biology that you're studying. And you need to be able to, defend yourself against the sometimes unexpected statistical consequences of having those aberrant cases in. So most sequencing quality control is discussed the way we did earlier, where people talk about, let's check the input sample to see whether we should sequence it. So biologists spend a lot of time on that. For some reason, they just hand the data files over when they're done. And they're like, I wash my hands of this, this is all. But the most informative QC analysis is actually post-run QC, where you're trying to see what did we get out of this whole assay system. There's a lot of places in the sequencing assay or microarray assay where things can go really wrong downstream of the input sample. If you just trust the sample implicitly based upon good input QC, all of those things will end up in your downstream data. So it's definitely critical to look at the data that you get out the back end of the assay process to see whether something has either crept through the initial QC downstream of that initial QC. So we'll talk RNA to even run this cDNA prep, this cDNA, production of DNA from our RNA, and is the stuff in reasonably good integrity? Although that second item, first one listed there, we actually are much more slack about these days because the assays work generally pretty well, even with significant degradation. So now we look for like only really severe catastrophic, it's just dust degradation before rejecting samples. So then, assuming the sample clears that pre-assay QC, we send it to assay with RNA sequencing or microarrays or something like that. The first thing we need to check out the back end from the transcript abundance matrix for each individual sample is, did I get the number of sequencing reads that I expect? So in general, remember, we target 10 million sequencing reads, but we can't control the exact amount of RNA that went in there, the exact efficiency of the enzymatic reactions, the exact efficiency of the robot crane, the exact efficiency of the pipette tip manufacturer. So there's always going to be these tiny little variations in how much RNA goes through each step of this assay process. And every once in a while, I'll tell this, robots get tired after a while, like their rubber O-rings get degraded. So every once in a while, a pipette tip will fall off. And so like you got nothing going on downstream of that, right? So you wouldn't know from input QC if that had happened. Now that's a rescuable situation. We can always solve that problem if we know about it. But most assay cores are like, yeah, that's going to be trouble. Just don't tell them. So there's all kinds of crazy stuff that can happen after the input QC. So the first thing you want to do is check and see, did I get the number of reads for this sample that I expect? There's always going to be some variation because of those imprecisions I talked about. Some people it's going to be 8 million reads. Some people it's going to be 12 million reads. Some people it's going to be 5 million reads. Some people it's going to be 2 million reads. And that's for real because their vein collapsed when they were, doing phlebotomy or something like that. We didn't get enough blood in the first place. We gave it a try. It's a much better idea to give it a try, see if it worked, and then throw it out if it didn't work, than to just assume that it's not going to work because, again, the things you throw out might be somewhat correlated with the things that you're interested in. So usually the first thing we check for is, did we get the general amount of data out that we expect? If not, if it's really, really low, like there's a point at which it's just catastrophically low. So if you're expecting 10 million reads and you get less than, certainly if you get less than 1 million reads, gone. If you get like 3 million reads, major flag for that. Let's run sensitivity analyses with this, and without it, it's in the range that's worrisome. If you got 5 million reads, generally quantitatively, 5 million produces almost the same thing as 10 million, except that 10 million is more reliable for these weakly expressed transcripts. So, you would prefer the 10 million all told, but you wouldn't want to throw out a quarter of your sample if you got 5 million reads, if it's going to give you decent data for the vast majority of your results. So there's not going to be like some clean, clear line in the sand where everything above this is good and everything below that is bad. But you are going to want to look at this kind of waterfall plot of like, okay, there's some of these things that are really super crazy poor, and I'm just going to eject those before doing anything else. And then there's going to be these yellow zone things I'm going to do a sensitivity analysis to knock out and make sure that they don't drive results that I get from otherwhere. The other thing that we look at, I see your question, is did we end up aligning as many reads to the human transcriptome as we expected? So remember I told you in most samples, about 90% of our reads, we can find a source in the human transcriptome. That's true in general. With dicey RNA input, you can, sometimes that walks down to 80% or something like that. Sometimes that's due to, bad sequencing chemistry or some degree of degradation. Sometimes in certain samples, it's due to infections. So like literally there are microbes growing in either in a person's blood sometimes, but more often they're like somebody left a plate out somewhere and there's stuff growing. Laboratories are hot. The stuff should all be frozen all the time. But I can tell you every once in a while, 96 samples, drop down in read counts. And so I'm just like, you guys can tell me whatever you want, but I know what happened there. So those are the two things you really want to pay attention to. Did I get as much data as I want? And did it seem to come from the human genome? If it didn't come from the human genome, either there's a technical problem or there's some weird biology going on here.
Are there negative controls that would actually have that they purposely put in the
Not so much anymore because those controls work right all the time. So that's the power of the base pair complementation. So they do that, but almost never do they actually, report it to you. They mostly do it as at the what we call runtime QC, which is completely, there's like a billion runtime QC parameters about how the sequencer is working. Most cores are not gonna hand you stuff that flunks runtime QC. So they'll, they do have controls for that kind of stuff, but it basically, in our experience, either it never goes wrong or they never tell us it goes wrong, one or the other. So. Any other? Yeah. Okay. All right, so the very first thing you want to check is, did I get out of this pipeline the data volume and, sort of results that I expect? And then, like a lot of people are like, woohoo, I'm off to analysis or something like that. I am a little bit more skeptical of this stuff because it's possible to have the expected number of reads and pretty decent alignment rates, but still have really aberrant looking data. And this is, again, what you guys are used to. You guys do, PC plots and all kinds of weird, sort of outlier detection things. I like at least a soft version of that kind of thing where you, I'm going to sort of skip that stuff just because we discussed a little bit more. But some people, will say, okay, and I alluded to this earlier, it's possible to get less reads than you expected or less alignment than you expected simply because we asked the sequencing core to not adjust the volume of samples for samples that have different, that have suboptimal input blood levels. If you adjust the volume of these things, the enzymatic reactions work differently and you get very different amplification, and therefore you get different stochastic behavior. So we generally tell sequencing cores, don't try to artificially inflate low RNA samples. If it's a low RNA sample, I'm happy to throw it out. Like I have no problem with that. I'm a methodologist, that's fine. Like I can just say, we just did not get valid data from this person. But I can't do that if you artificially inflate the number of reads, so I can't tell whether this stuff is reliable or not. You can always make more reads with with fancy chemistry, but all you're doing is multiplying dust. So you're just creating, 500 copies of gibberish as opposed to, and I can't, once you've created that much gibberish, it's much harder to tell whether it's gibberish or whether it's true stuff. So we tell people, if you get suboptimal RNA, take a shot at getting us the data. But if it doesn't work, that's the sample's fault. It's not your fault. We'll still pay you for the assay, all this other kind of stuff. Because otherwise, they're going to try and make it look like they think you want it to look. And it will actually shoot you in the foot because it's very hard to figure out when stuff is wrong. So it's much easier to do that. And so in some scenarios, we'll say we got less reads than we expected. And it turns out that in many cases, you can actually either rerun the assay, or most of the time they create way more cDNA than they actually sequence. So you can actually just pull an aliquot of cDNA out of the freezer and say, hey, let's just redo these 5. But those 5 have to have a big yellow caution flag attached to them for the rest of your analysis, because they're going to have somewhat different stochastic behavior. So you're definitely going to want a sensitivity analysis to do those things. But it is possible, if you catch it soon after the assays are performed, before the sequencing core has thrown away your cDNA, basically, you can actually get more sequencing precision if you ask them to. You'll have to pay for it, but it's actually not that terrifically hard to do. So that's one sort of possible response to this. The other thing is, if you don't have blind faith in technology, or if you think that there's other kinds of stuff that might imperil the validity of your analysis besides technical validity, you can do these endpoint quality control analyses that are based essentially on consistency. So one super easy version of this is the profile correlogram, which I'll show you guys in a minute. Another version is what we call ground truth analyses, where we're like, I want X and Y linked gene expression to correlate with self-identified sex. It doesn't have to be right all the time. I'd like it to be right like 90, 95, 99% of the time, but not the end of the world. But I don't want it to be systematically wrong, which happens all the time because people misalign data sets. So there's a whole bunch of terrifying places in this whole thing where people are cutting and pasting stuff from Excel into whatever Illumina software. And if they get one cut and paste operation wrong, it's like a complete random correlation between sex and everything else. So that is a great thing to detect before you start analyzing data, right? So it's like very fine idea to go and do some ground truth analysis to just make sure that those kinds of misalignment errors haven't happened. And then, once those are good, it turns out, most of the time, things are fine, and you would have been just as good not doing the endpoint QC, but it's definitely good to know about, especially on the occasions when people have leukemia. So I'm going to skip through some of the QC stuff. We used to, in microarray world, worry about these fluorescence distributions and throw out things that had bad fluorescence distribution, which usually had known chemistry reasons. But that's actually not so much of a problem for sequencing, especially now that we think I should have about 10 million reads. And if I've got 100,000 reads, this is catastrophically messed up. And so I'm going to throw it out. If I've got 5 million reads, it's not great. So, flag it. If I've got 2 million reads, yeah, probably really throw that out. If I've got 8. So, if you've got a sense of what you should be getting, then that mostly replaces this more complex distributional stuff. But in general, regardless of whether samples are really bad, you're still going to want to clean up that noise that comes from different numbers of reads being acquired for different samples. So the typical way to do this is very simple. You just express the reads not as raw counts, but as counts per some arbitrary denominator. The typical version of this is counts per million or transcripts per million, or so-called CPM or TPM values, which just expresses these things as percentages of a million instead of percentages of 100. So the number of reads that align to a given gene divided by the number of total mapped reads, not the total number of reads you acquired, but the total number of reads that you could find a location for on the human transcriptome. So it's really counts per million mapped reads, but that's a little bit less convenient acronym there. So that's the normal normalization, or the first stage normalization. Some people, for various reasons, and this is a little bit trickier for us, for most of us in like social population behavioral health, you probably won't need to do this kind of thing. But for people doing biological studies, this may turn out to be more important. But sometimes you additionally want to what we call post-normalize based on holding constant some assumed set of what we call reference genes that should be stably expressed. The trick is that actually with sequencing, we've discovered that that should almost never holds true. There's almost always some situation where the things that we call reference genes or housekeeping genes that should be invariantly expressed are nevertheless ramped up by some interesting biological process or suppressed by something else. So it's hard to find the reference genes, but people have come up with lists of these things. So we should be able to do that. Usually, when you're going to want to normalize to reference genes is when you tried analyzing the data in counts per million or transcript per million metric, and you looked for differences in expression, and you found 2 upregulated genes and 2,000 downregulated genes. That often is due to some what we call dark matter garbage in the total million reads, where there's some sort of big clump of transcripts that is expanded and therefore is contributing to the total million mapped reads denominator. So it's usually not the numerator that ends up creating these weird things. It's a big expansion of some transcripts in the denominator that ends up essentially artificially increasing the counts per million relative to the distribution that would be there if that population of transcripts hadn't expanded. Often we don't know why that population of transcripts hasn't expanded. There's all kinds of weird stuff, like hemoglobins are some of the most highly expressed transcripts, and it's often the case that there's huge expansions or contractions of hemoglobin transcripts for reasons that probably there's an understandable biological explanation for, but the patients are healthy or the people are healthy. Can't see the reason a priori to exclude them as invalid, but there's this huge gyration in the denominator that we like to think of as a kind of a stable population. So when that happens, normalizing to reference genes that aren't subject to that sort of dark matter, like we don't know what they are, we don't know why they came this way, we don't know why they're expanded, but they're throwing off our denominator. If you normalize to reference genes, where we take, let's say, 10 or 11 genes that are believed to be relatively constantly expressed across cells, and we basically, sort of normalize by the whole constant that population of genes as transcripts per million, and then see what we end up by applying the same normalization factor to all of the other genes. Okay, so the one thing I did want to show you is this sort of parallelogram approach to identifying aberrant samples. This is a pretty easy thing to do. There's lots of fancy outlier detection stuff that you can do, but one really simple thing to do is to just take the columns of that transcript abundance matrix and compute correlation coefficients between them. So you can compute the correlation coefficient between A and B, column A and B, column A and C, column A and B, so on and so forth, and clump those correlation coefficients in, and then just color them heat plot style. What you'll typically see is generally reasonably high correlations. If you're working from, gold standard, lots of RNA situations like pathogen tubes or something, the cross-sample correlations will typically be 0.85, 0.9, something along those range. But every once in a while, you'll come to a sample that has an average cross-sample correlation of like 0.6 or 0.2 or some crazy thing like that. And again, you'll want to go back and check, is this coming from a sample? Usually this stuff comes from samples that have low transcript counts altogether, and so their transcript distribution is mashed in weird ways. Or they come from samples that have like some weird, sort of biology going on, like, again, leukemia, you've got a bunch of one kind of white blood cell, way more than its normal prevalence or something like that. And so you've got some dramatic, sort of change in the population structure of this cell pool that's creating the RNA pool that you analyze. But it's really easy to pick these things out if you do this heat plot coloring, because they will produce, sort of I now do it, sort of red high, blue low. And so you'll get these blue cross patterns that very strongly mark the samples that look different than everything else. So then you check and see, are these really low transcripts per million, or really low total transcripts, really bad alignment rates? If that is true, generally we get rid of them. If it's not true, if it looks like the right amount of data and reasonable read alignment, so kind of technically okay, but still aberrant, that usually points towards something weird biologically. And then you have to start answering this personal philosophical question about, is my question sort of a nomothetic question about general relationships, in which case having this aberrant sample, runs the risk of sort of distorting these generalizations, or I have no affirmative grounds for excluding this sample. I'm going to have to keep it in because it was validly sampled from the population of interest. So that's going to be kind of up to you. But at the very least, you might go back and say, okay, do we know anything else about sample G there that would, is this person sick? Are they missing all their appointments? Is there anything that would lead us to believe there's a more general problem here? All right. I promise only about maybe 15 more minutes, and then we'll be off to lunch. Question?
Obviously, are there a list of treatments that we would maybe want to preference apply for average?
There's not like a simple list. So the question is, if you see an aberration like that in the post-run QC, what are the things that you would look for as likely explanations? So hematologic problems of some sort or another are definitely one. Another one is medications that's strong. Like if you're doing a study of blood, it's going to be meds that affect blood cells in particular. So the major actors there are anti-inflammatory medications. So this includes things like glucocorticoids, steroids, as well as these antibodies against things like TNF or IL-6, these strong biological response modifiers, they call them. So you might want to look for drugs like that, methotrexate, other kinds of agents that sort of act on that system. You also want to look at just like their general medical, sort of like their pharmacy list, for lack of a better term. Most drugs that we take don't have very big effects. So there's relatively modest effects of drugs that are highly used, like let's say, birth control pills, or oddly enough, non-steroidal anti-inflammatory drugs that people take when they're not having major inflammation. They don't have huge effects. Antidepressants don't seem to have big effects. So thankfully, most things don't. To some extent, the answer to that question depends on the specific types of genes that you're looking at. So for instance, for these stress responsive genes that we often look at, whether you're taking a beta blocker makes a difference, and actually which kind of beta blocker it is. If you're taking the old style propranolol beta blocker, it will definitely make a difference. If you're taking the so-called selective beta blockers that cardiologists now preferentially prescribe, then it actually won't make much of a difference. So that only makes a difference for a restricted set of genes. But for white blood cells in general, it's mostly the things that docs give to people to mess with your immune system. So usually it's pretty intuitively apparent. And my impression is that most people are more worried than they should be about most medications. It's most medications, there's actually surprisingly little impact. Yeah. Well, you should be so lucky as to have that problem, but I would definitely worry about a correlation of 1 with a sample that is not the same, that's literally like, if we just re-assay the same physical sample, we're not going to get a correlation of 1. So there's no magic upper limit. In fact, we just, again, we're just like, this is just not a problem, right? I mean, it's like we're constantly trying to drag ourselves out of the mud. Like, can you hyperventilate after breathing mud for years? Yes, you probably could, but it's like you're still happy to be breathing. So we don't really see anything that would ever lead us to exclude on the high end. One of the places where people have thought about this is in genetics, where they assumed that the correlation between, let's say, siblings or identical twins should be, really high. Ironically enough, that there's very, very little, genetic structure to this kind of stuff. So there's like, it's in big samples, it's discernible. You can find it if you're looking for it really hard, but there's relatively little prevalence of that issue and relatively little explanation for it are there other than just random chance. All right, so the last thing I'll do right before lunch, everyone's like, wait, you're doing statistics right before lunch? But this is because this is like throwaway easy. So, and we'll go over this all again and again. So think of this more as like an advanced investment in what we're really going to be doing after lunch. But basically, how would we attack the analysis of that transcript abundance matrix? So before lunch, what I'm going to talk to you about is what we'll call the classical genetics approach. And then after lunch, we'll kind of expand the scope of the kinds of analyses that get done and describe where genetics approach sits within that and how it's different from some of the other approaches that might make sense. But basically, the general stream of analysis is, one, try and get rid of, normalize away as much purely technical variability as possible so that we can simplify the analysis and not have to include essentially regressors to take all that stuff into account. So we talked a little bit about that. Sample quality control, knocking out weird, aberrant, outlier, provoking samples before they can mess you up is a good idea. Normalization to try and stabilize the sort of the purely methodological variance that comes from some people gave more blood, or the pipette tip took a slightly more volume in this sample than in that, something like that. So the next thing that we typically do is try and stabilize biological variability. This is in the kinds of analyses that we do not quite as catastrophically problematic as it is in other scenarios, but in like studies of free-range human beings, rarely are we doing crazy things like feeding them a lot of bacterial cell wall to ignite their inflammatory response. But some people do that kind of stuff. And so they do need to do this stuff because they are going to produce 10-fold or 100-fold or 1,000-fold changes in gene expression. But most of us who are just looking at people under basal conditions, there's nowhere near that much extreme biological variability, but nonetheless, it's customary to do this, and there's nothing to be lost by doing it. So usually we stabilize biological variability by custom in the RNA analyses. This is done in a log2 metric, in part because the intuition of biologists who were starting this stuff, before the social transcriptomics world came along, they sort of thought of twofold as a meaningful difference. Twofold is a really high amount of change for most social or behavioral influences, but they were used to, again, dropping bacteria or neurotransmitters or something like that. And so they were producing very big responses in these highly controlled experimental settings. So they thought twofold, that's a big effect in their world. That's more than you would usually see due to noise alone. So they kind of settled on log base 2. And so the advantage of log2 transformation is that you get numbers for meaningful ranges of variation that are kind of visually appreciably different in this kind of log2 metric, but they're not as sensitive to low, let's see how to think about this. It's basically, it's good for visual data matrix inspection as well, because it doesn't create a bunch of numbers that vary only in the third or the fourth decimal point. If you use a log10 transformation, you're forcing the meaningful variance farther down the visual field where it's harder to pick up. So log2 also has the advantage of being a little bit more clearly discernible. I'll show you this stuff in a minute. One of the problems, though, that you guys will recall from earlier is that RNA data, especially from sequencing, often has zeros in it. So when you go to try and log transform the zeros, you're not going to get appreciation from your statistics package. So you need to substitute in some kind of, replacement for zero in there. And the custom generally is to put in 1 transcript per million as the lowest observable value. Some people push that up higher and will say like 10 transcripts per million or something like that. But replacing zero values with a 1 or a 10 or something like that is essentially the normal typical solution to this kind of thing. If you replace with a too high a number like 10 or 100 or something, you're going to squash a lot of low range variability that actually might be real. If you put a value noticeably less than 1, you're going to be allowing what we believe to be mostly noise results to achieve, twofold or more effect sizes. So part of the reason people select 1 transcript per million is because that is about the level at which you can see what you call qualitatively consistent counts in replicate analyses, as they were talking about earlier. If I run the same assay on the same physical sample of RNA, I'm going to get 10 counts this time and 6 counts the next time and 12 counts the time after that or something like that. So, that's broadly consistent. You're not getting 10 this time, 100 that time, 0 the next time. So it's about 1 transcript per million where you get reasonably what we would think of as like qualitatively consistent results. Below that, the variation is too high. So if you go below 1 transcript per million, you can easily get, counts that produce 2 or 3, 4, 10-fold effects, but are, the difference between 1 count and 10 count or something like that. So as long as you're acquiring 10 million reads as your general target, or even 5 million reads, 1 transcript per million is about the right thing. If you're acquiring many more, you can bump that up. If you're acquiring many less, you might want to, you should still probably keep it at that, honestly, because the counting mechanism is just too imprecise at the low end. So once you've transformed to stabilize biological variability, then it's pretty straightforward. What they do is they go through gene by gene, like every row of that data matrix, and they'll fit some kind of statistical analysis to it. So some linear model, generalized linear model, whatever makes sense for your particular purposes. And they're going to pull out a regression coefficient that summarizes the magnitude of difference in log2 messenger RNA abundance as a function of this predictor. So this could be like a t-test between 2 conditions, or analysis of covariance controlling for everything, and looking at the effects of SES or something like that. So that regression coefficient, usually people's instinct, and this is especially true of statistical geneticists, they really love p-values, and so it will almost always pull a standard error and a p-value out of this. But those p-values are not very trustworthy because there's 20,000 of them. So if you run at least 20,000, maybe 50,000, if you run that many, repeated tests, you're going to get hundreds of genes statistically significant purely by chance alone. So generally, the statistical response to this has been to control for multiple testing somehow. There's a wide variety of ways that people can do that. The consensus in the RNA field has mostly been around false discovery rate testing because it's not quite as insanely stringent as Bonferroni correction. But, and it does, theoretically, it solves this problem. Practically, after lunch, I'll show you, it doesn't work. Like it's completely not right about what people's intuitions suggest it's about, which in their defense, FDR people never really said, I promise you a replicable result. They said, this is the number of false discoveries that you're making. But people are like, but I want a replicable result, not false discoveries. So, so that, we'll go through later. But suffice it to say, very early, people discovered that not all p-values of less than 0.05 or less than 0.005, if false discovery rate corrected, are the same. Like a p-value, a very small p-value computed on a highly abundant gene transcript is much more likely to replicate than a very small p-value computed on a transcript that is mostly zeros, but a few ones or twos. It turns out if you run enough of those things, you'll get some things that clear your FDR threshold, right? You'll get stuff that is nominally statistically significant even after correction for false discovery rate, and it will not happen next time because these 0, 0, 0, 1, 0, 0, 0, 10, 0, 0 things, they're mostly noise. So it's very easy to capitalize on chance if you ever press a sort button. That's what they do. They capitalize on chance. They'll find true stuff, but they will, if there's any chance, the sort button will find it. So that's what they discovered when they started analyzing RNA data. So they moved to this process of taking into account both the p-value or the false discovery rate corrected q value and the effect size, which essentially amounts to the regression coefficient. So they now mostly analyze data in terms of this sort of requirement that you have both statistical significance and some kind of substantive significance in terms of some, effect size that is believed to be of biological significance or import. Intuitively, that seems to be like 1.25 fold or 1.5 fold or twofold. If you use 1.6 fold, people will get very upset. If you use 1.5 fold or twofold, they're like, oh, this is fine, that makes perfect sense. So there's all kinds of opportunity to optimize this statistically, which nobody wants to do because they get very nervous when a statistical tool has optimized their threshold. So the reality is people choose some kind of what seems to be intuitively reasonable, sort of real world quantitative effect size in the form of the regression coefficient, like something that comes out to a 25% difference or a 50% difference or something as a screening threshold. So usually they will not allow just an FDR curated gene list. Some people will sometimes go along with that, but people who are actually kind of serious about is this a reliable, replicable result will generally kind of balk at that. So the solution that they come up with for displaying this stuff is called a volcano plot. And it's a volcano plot. Each dot corresponds to one of the transcripts assayed. They're arrayed by negative log 10 p-values. So the results that are most statistically significant are at the top of this plot. In other words, the log 10 p-value, which would normally get small. If they negative it, it will get large. So highly significant stuff is high on the plot. And then on the horizontal axis is the, I'm not quite sure what they wrote there, but basically the upshot is that's the effect size as divined from the regression coefficient. So I think that's like, is it log 2? I can't see it. Yeah. So then the 2 is actually a fourfold effect. The 4 is 2 to the 4th. Basically those are pretty big effect sizes. As you can guess, that did not come from a social or behavioral science study. That came from some crazy, sort of experimental laboratory biology. But you can see they put some vertical bars at plus or minus, it looks like about 1, maybe 1.5 or something like that. And then a horizontal bar at, I can't even read this thing, but 2. All right, so that's like 0.01. So the stuff in this volcano plot that is deemed to be reliable or more reliable is the stuff that is higher up and more to the right, red, or more to the left, blue, as in downregulated. So you often see these volcano plots with these quadrants carved out of them, either by some theoretical calculation. If you did the false discovery rate correction, usually false discovery rate bounces the statistical significance threshold by a factor of 10 to 100. So it's probably kind of, false discovery rate threshold would probably be about 4 on the vertical axis. And the key point is that you can see a lot of stuff that is above the gray horizontal threshold, but not greater, not very far to the left or right of 0. What that is really just telling you is you can get very small p-values from very, very small effect sizes, and they're very unlikely to replicate compared to the same small p-value, or absurdly enough, a much larger p-value, but a much larger effect size. So that is the part where people eventually realized you can't squash all of the information about RNA abundance into any single one number representation. You can't do it with p-values because they keep dredging up things that are statistically significant, but practically of almost no magnitude. And you can't do it just by, sort of looking at, effect size alone. Although if you were forced to choose one or the other, actually effect size does pretty well these days, as long as you use a reasonably large effect size for the situation that you're working in. So one of the other things that people often did was kind of a global signal test, where they said, okay, I've got a distribution of p-values essentially summarized by the sort of the vertical axis, the spread of data there. Maybe I can just see, did anything happen in this experiment at all? So I can plot the distribution of p-values, and in essentially a close to null hypothesis or null hypothesis scenario, usually that distribution of p-values is going to be a kind of a regular uniform rectangular distribution. You'll still get some statistically significant p-values by chance alone, but under the normal theory of kind of statistical testing, you'll get, under the null hypothesis, a uniform distribution of p-values. And in fact, that's often what you see in variables that don't have a very large influence on, let's say, blood cell transcriptome profile. So for instance, there's very little difference in probably from Ad Health, between the Hispanics and the reference Caucasian group in terms of the distribution of blood cell RNA abundance. But sometimes there are, meaningful effects. So this is, I think, also from Ad Health, if I remember correctly, where we're looking at the distribution of p-values for body mass index. And you can see there is a very large stack of very small p-values spiking up on the left there. So in the global signal test, what they do is they use a null hypothesis, which you can kind of calculate from the number of tests that you're doing and their supposed individual false positive rate. And you can calculate out that null hypothesis, which is illustrated in the horizontal dashed lines in those two plots. So in the plot on the right, which you'll appreciate, has a greatly expanded vertical axis to deal with that big pile of significant results. But the dashed line is actually at the same place in both graphs in terms of like the vertical axis number. But you can see there's this huge stack of small p-values on the left, and there's a deficit of large p-values on the right, which is where a bunch of genes migrated away from as they showed their statistical significance in association with BMI. For most of the stuff that we look at, it's going to end up looking like this. So this is in Ad Health. That's kind of what the poverty p-value distribution looks like. So the good news is your eyeball will tell you there's probably something going on. Looks more than chance stack of small p-values. But sadly, when you run the actual calculations, they'll say, nah, there's not enough of these things to decisively reject the null hypothesis under standard false discovery rate statistical theory. So it's not a bad idea to make plots like this to just give yourself a sense of, are you here, which is going to pull, or are you here, where you're like, switch my topic for the future or something like that. But rarely are we so lucky as to have stuff where it's obvious and incorrigibly, statistically significant that our variable is doing the same. So maybe you'll do a global signal test. I personally wouldn't do the test. I haven't done these tests in years. But making the graph is actually not a bad idea, just to get a kind of an intuitive sense of effect size. But oddly enough, and we'll talk about this more after lunch, the most replicable results don't come from selecting on p-values. They actually come from selecting on the effect size, on the regression coefficient. And that is a funny bonus from the amazing reliability of the measurement infrastructure we have. This was true, actually, oddly enough, we discovered this for microarrays. We were using machine learning tools to find these thresholds by just saying, can I predict from 20 replicates whether another 10 replicates will show an average, twofold effect or greater or something like that. So as you might imagine, machine learning algorithms are like navigating around in this two-dimensional space of, regression coefficient and p-value or q-value or whatever you wanted to do. And by the time Illumina started making microarrays, the machine learning tool would, if I was asking it to find me genes that were going to be a twofold effect or something like that, or even a 1.5-fold effect in future replicates outside the prediction sample, often the machine learning algorithm would just ignore the p-values. Like this stuff is not useful information. As long as you're setting the threshold, reasonably high, like if you're setting a threshold at 1.5, given the noise structure in Illumina, it's not like you never make a mistake. It's just you make far fewer mistakes than your intuitions would suggest if you do this, and you suppress spurious noise. You do that flooring that we talked about in transcripts per million. So we'll talk more about this later, but the upshot is how you do this analysis depends a lot on what you're trying to write when you write a paper downstream. If you're trying to say, I've pulled out this gene, IL-1 beta, and it is significantly associated with my predictor of interest, P equals something, then actually you do need to do the false discovery rate corrections. In other words, you can't do a gene by gene interpretation, a single gene at a time. You can't come up with a list of genes and say, this is reliably correlated with my variable of interest. This is. Like if you're doing single gene at a time interpretations, you absolutely need to do it this way. But it turns out most people are not doing that anymore. Most people are doing gene set analyses where they try and interpret sets of genes and their properties, not individual genes. And there, they're usually not actually trying to ascribe statistical significance to individual genes. They're trying to ascribe statistical significance to latent factors or conceptual entities or common characteristics that these genes share. And there, if you do statistical testing at the level of individual genes, you inadvertently create a tremendous, almost insurmountable bias in the information that's going into your next layer of analysis that actually prevents it from getting the right answer. So this is one of the sort of more curious developments here, is the surprising power in the context of good, measurement, of just letting the point estimates be the point estimates and progress on if your ultimate interest is not in the individual genes, but in what they reflect in terms of things like inflammation or antiviral biology or atherosclerosis or that kind of stuff. So we'll talk more about that after lunch. So one last point, DNA genetics, it's not a great idea to call RNA DNA genetics because it triggers a whole bunch of concerns that actually aren't relevant in RNA related to individual identification and ethics risks, broadly speaking. But there's one exception to that, and that's the raw sequencing data. So the raw sequencing data does pick up polymorphisms. And so there is enough information in most raw sequencing data to identify individuals if that information was made public. So in general, the count matrix, there's nowhere near enough information to identify individuals. So that stuff, there's no particular security risk. But most of us generally, resist the idea that we should be posting individual level raw sequencing data because you can clearly use it for, re-identification purposes. So just be aware that while you shouldn't characterize RNA as DNA genetics in general to avoid misinterpretation, you do have one particular domain of DNA style risk at the level of the raw sequencing data. Okay, so that's all we got for now. We are going to go to lunch. I have two things that I've been asked to let you know about lunch. One, you need to pick up a voucher from at the door. So Teresa will hand you a voucher as you go out. Those of you who are staying in the hotel probably already got a voucher, so feel free to take two and then eat twice as much lunch or whatever you want to do. The other thing is Teresa needs a dinner count. So and we'll be back here an hour from now. Is that right? At 1:30 or 2? I can't remember. 1:30, but thank you.